Hello everyone, good morning. Am I visible? Am I audible? Are you people there? So good morning everyone. So let everyone join. I will start the class in a minute. Okay, let everyone settle. Can someone write in the chat box if I'm clearly visible, audible? You can see my slides also. Yes? Can you write down in the chat box? Good morning. Good morning, Mona. So is everything is good to go? Good morning everyone. Everything is good to go. I mean visibility, audibility. Yes. Okay. Thank you. Thank you for confirming. Thank you so much. So that's what I want. So let everyone join and we will start today's marathon. Right? The complete bacteriology in one shot. We are going to complete today. Okay? Complete bacteriology. One shot. Today, the complete day. Okay? So I'm good. Hope you all are also doing good. And today we are going to complete entire bacteriology. So we have started microbiology. Yesterday in microbiology, we have completed two sections. We have completed entire general microbiology. I guess we have completed entire general microbiology, yes? So yes, and we have completed the immunology apart from two topics which are pending. Okay, so two or three topics are pending in the immuno. Rest immuno is also completed. So today I am targeting entire bacteriology in the most simplified manner. So the strategy is that I have provided you the detailed notes. The detailed notes are something 110 pages. So what is the page number of bacteriology? Can someone tell me in the detailed notes? So I will share with everyone. I want you to open that detailed notes also. So what is the page number? Otherwise, I have to open. What is the page number of bacteriology? Yes. Can anyone share the page number of bacteriology in the detailed notes? It's page number 47. It's page number 47. Please, everyone, open page number 47. Right, number one. And number two, I have provided you the charts also, which are known as master tables, the comparative charts. The master tables are also with you. So in the master tables, it's starting from page number one, or what is the page number? Let me see. So yes, it's from page number one only. I guess it's from page number one. So Staph aureus bacteria. So you open that also. So I want you to have an eye on both. Now it's up to you. Whatever I am teaching you is this, the detailed notes. But whatever you have to learn in the end, the summary is given in the master charts. Now you want to follow this or that, it's completely your choice. ठीक है, मैं पढ़ाऊंगी डिटेल नोट से, लेकिन तुम्हें अंत में याद मास्टर चार्ट ही रखना है. अभी मुझे पढ़ाते पढ़ाते तुम्हें मास्टर चार्ट फॉलो करना है या डिटेल नोट्स फॉलो करना है, इट्स योर चॉइस. लास्ट में यू हैव टू रिवाइज मास्टर चार्ट्स बिफोर गोइंग टू योर एग्जाम. ऑल द क्वेश्चंस आर कंटेन्ड इन दैट. समझ में आई क्या स्ट्रेटेजी? Everyone got the strategy? Give me a thumbs up. Can we start the class? You got the strategy? Can I start the bacteriology? Everyone with me. Everyone. And I want everyone to be interactive, energetic, awakened during the class. मेरी क्लास में सोना अलाउड नहीं है. ठीक है? So can we start? Say yes, people with full energy. Okay, so let's start. So I'm starting with bacteriology. Listen now. Based on the gram staining, we have seen yesterday we have two types of bacteria. What are the two types of bacteria based on gram staining? The violet are gram positive and the red are gram negative. We have already seen that yesterday. Okay, based on the shape also, we have two types of bacteria. The spheres, can you see the spheres? Spherical. Spheres are cocci and the elongated one, lambe jo hain, elongated. These are known as bacilli. So based on the gram staining, two types. Based on the shape, two types. Let's combine the two classification. We have four types of bacteria. So let's combine. We have four types of bacteria: gram positive cocci, gram positive bacilli, gram negative cocci, gram negative bacilli. So this is the ultimate classification of bacteriology that we have seen yesterday also, and we are revising and starting the bacteriology. So we have four classes of bacteria. What are the four classes of bacteria? Gram positive cocci GPC, gram negative cocci GNC, gram positive bacilli GPB, or gram negative bacilli GNB. I want all of you to see the color and shape. Yesterday also I taught the same. See the color and the shape. You will understand the name. Can you see these two are cocci and these two are bacilli? You can see. These two are cocci. See the color. The color is violet, it's gram positive cocci. The color is red, it's gram negative cocci. You can see these two are bacilli. These two are bacilli. The color is violet, gram positive bacilli. The color is red, gram-negative bacilli. Now you have to learn this classification. Yesterday also we have seen, I know that. I'm just revising you to tune up. And after that we will start the first bacteria. One by one we will start it. Till end, Mycoplasma will be my last bacteria today. And Staphylococcus is my first bacteria today. So in this sequence only we are going to conduct. Okay? So the 4 categories are in front of you. So in gram-positive cocci, we have only 2 bacteria. Can you tell me the names? Yesterday we have studied. Naam batao 2 bacteria के. Fatafat, Staphylococcus, Streptococcus, SS. So we will start with SS. है ना? It will take 1 hour 30 minutes to complete SS. These 2 are big bacteria. 40 minutes each. The big bacteria are there. ठीक है? After that, the next 2 is Neisseria and Moraxella. The gram-negative cocci, it's Neisseria and Moraxella, only 2. I will hardly require 30 minutes here or 20 minutes. It's very small. है ना? So first we will finish gram-positive cocci, SS, and then we will finish gram-negative cocci, NM. ठीक है? ये 2-2.5 घंटे में हम इतना finish करेंगे. Then we will come on gram-positive bacilli. There we have 6 bacteria. Can you tell the names? A, B, C, D, L, M. What is the full form, people? A for actinomycetes, B for bacillus, C for clostridium, D for diphtheria, L for listeria, and M for mycobacteria. So these are the 6 bacteria which are present in gram-positive bacilli. The 6 bacteria. Remaining all will fall in the last category. So we don't learn the names. जो भी बच गया, except these 10. SS को छोड़ के, NM को छोड़ के, and ABCD LM को, remaining all will fall in the last category. So we will see the last category. So this also required 2 hours. So my target before lunch is to complete these 3 categories. And after lunch, we can complete the gram-negative bacilli. Or we can start that also. Okay. You already have the notes. You have the long notes, elaborated notes also. And you have the charts also. You have both. You don't have to write even a single word. You don't have to write. Mind my words. You will not be able to write with my speed. You will be able to mark. So take your notes. You have hard copy, you have soft copy, whatever copy you have, open that. Either this or this, it's your choice. You want whatever I am teaching, dipto you want, you open the long. You want ki nahi, jo learn karna hai, summary batao, you open short. Whatever is your choice, but mark it. Don't try to write anything. Otherwise, you will not be able to cope up with the speed. It's an advice. To follow or not, it's your choice. So let me start with the first category, gram-positive cocci. Before starting that, one by one, we will start. So you got the plan, So what is the plan? The plan is we are starting with this category, then we will move on this, then we will move on this, and then finally with this, and we will end with this. So in this 5 section, we are going to complete entire bacteriology today. Okay, listen my point. Before starting that, I want to tell you the morphology of all bacteria together. एक ही बार मैं बता देती हूँ हर बैक्टीरिया वाइज आई विल नॉट टीच यू द मॉर्फोलॉजी ठीक है मॉर्फोलॉजी मींस फॉर एवरी बैक्टीरिया यू हैव टू टेल मी 3 थिंग्स जो भी बैक्टीरिया पढ़ा रही हूँ तुम्हें 3 चीजें बोलनी है वेदर द कैप्सूल इज प्रेजेंट और एब्सेंट उस पर्टिकुलर बैक्टीरिया में वेदर द स्पोर इज प्रेजेंट और एब्सेंट इन दैट पर्टिकुलर बैक्टीरिया एंड वेदर दैट बैक्टीरिया इज मोटाइल और नॉन मोटाइल you have to tell me just suppose first bacteria I'm going to teach you I'm starting with staphylococcus so for staph aureus you have to tell me capsule present or absent spore present absent motility present absent for staph they all are negative no capsule no spore no motility the next I will teach you pneumococcus so for pneumococcus capsule is positive but other two are negative so if you learn the combination you will become mad हर बैक्टीरिया के लिए you can't learn negative positive positive various combinations are there you will become mad the advice is that learn 9 capsulated bacteria rest all are non capsulated learn 4 spore forming bacteria rest all are non spore forming and learn the 12 6 plus 6 bipolar 6 by peritrichous 12 motile bacteria rest all are non motile and apply your particular bacteria the 3 mnemonic I want you to learn the 3 mnemonic yesterday I have taught you the 3 mnemonic you people were present yesterday कल थे क्या क्लास में you people were there if you were there you may be knowing what I'm talking about so there are only 9 bacteria we will revise the mnemonic and move on there are only 9 bacteria which have capsule only 9 say the name of the 9 what is the mnemonic is yes very killer bacteria have pretty nice capsule you can count they are 9 B is double they are 9 in number I'm not reading the full form you just read it yesterday I have read it ठीक है there are only 4 bacteria which form the spore only 4 name it है ना ये mnemonic से याद करना है इससे करो पसंद नहीं आया कोई और mnemonic बना लो if you don't like this mnemonic make some other but you have to learn देयर इज नो एस्केप, लर्न तो करने पड़ेंगे. देयर आर 4 बैक्टीरिया व्हिच फॉर्म स्पोर. सो नेम द निमोनिक, है ना. इट्स बीएससी केमिस्ट्री. BSc chemistry. Learn the full form. I'm not reading it because yesterday I have read it. So it will take time. You just read it at your end. Just a second. Okay. And there are 12 bacteria. 12. Total 12 bacteria which are motile. 6 by peritrichus, 6 by polar flagella. You know the two mnemonic, cute baby sleep, very polar solution HCl. Okay, just suppose I'm teaching you my first bacteria, Staph aureus. I will start with Staph aureus. First bacteria, यही पढ़ाऊंगी मैं अभी. है ना. Does Staph aureus come in this mnemonic? Say no, ma'am. Is it coming in this? Say no, ma'am. Is it coming in these 12, 6 plus 6? Say no, ma'am. So you will say, ma'am, Staph aureus is non-capsulated, non-spore forming, non-motile. समझ में आई क्या बात? The next I will teach you pneumococcus. So you will say, ma'am, pneumococcus is coming here. So it's capsulated, but it's non-spore forming, non-motile. In other two, it's not coming. For example, I will teach you Bacillus anthrax. So you will say कि ma'am, Bacillus is coming here. है ना. Bacillus is also coming here. ये रहा Bacillus यहाँ पे भी है, but it's not coming here. So it's capsulated, spore forming, but non-motile. So my point is that morphology of all 30 bacteria I taught you together. I will not teach you morphology of even a single bacteria. You apply that bacteria in the mnemonic. भाई mnemonic में apply करके देख लो. इस mnemonic में आ रहा है क्या? इसमें आ रहा है क्या? इसमें आ रहा है क्या? आ रहा है तो है, नहीं आ रहा है तो नहीं है, खत्म बात. How many of you got my strategy? मेरी बात समझ में आई क्या? How many of you? If you have the doubt, please ask. है ना. So I will not teach you morphology individually even of a single bacteria. I have taught you morphology of all 25-30 bacteria together in one shot. This is a way we see. है ना. So we will not see the morphology. एक बार में पूरी हो गई. The 3 things in the morphology. Capsule, yes or no, you can answer. है ना. Spore, yes or no, you can answer. है ना. And motility, yes or no, you can answer. If you know the 3 mnemonic, 9 plus 4 plus 12, you should learn this mnemonic. The mnemonic should be at the tip of your tongue. नींद से भी उठा के पूछना तो याद होना चाहिए. ठीक है, शुरू करें. So let's start it. Can I start with the first category? Are you people with me? Can I start gram positive cocci? Yes. Let's start with gram positive cocci. ठीक है, I'm starting with gram positive cocci. ठीक है, with this orientation, there are only 2 bacteria we have to study here, Staphylococcus and Streptococcus. I'm starting the staph and the strap, the 2 bacteria. है ना. Now you tell me, I'm teaching you gram positive cocci. ठीक है, there are only 2 bacteria in this category, staph, Staphylococcus, and strap, that is Streptococcus. What is the morphology of both? By morphology, I mean what is the diagram of both of them? You will say, ma'am, both of them are cocci. That's why both of them are sphere. This is also a sphere. This is also a sphere. And both of them are gram positive. So both of them are violet. So let me fill violet here. Let me fill violet here. My first question to you, how to differentiate the two? Looking in the diagram. Is there any difference in the diagram? No. This is also violet. This is also violet. This is also sphere. This is also sphere, because both of them are gram positive cocci. My first question, how to differentiate the two based on the diagram? Based on the diagram, there is a way. Yes, there is one way by which we can differentiate. Kavita, Saeed, there is a way. Let me tell you a way. ठीक है? I agree the color is same. I agree the shape is same. But the point is that arrangement is different. The Staphylococcus are in clusters and Streptococcus are in chains. See. Beautiful diagram. This is staph. The staph is always in cluster. है ना? And this is strap. The strap is always in chain. Always in chain. Appreciate the chain. Appreciate the cluster of the grape. अंगूर के गुच्छे के जैसे होते हैं. है ना? So my point is that the arrangement is different. This is known as arrangement of the bacteria. That is different in the two. See the arrangement here. See the arrangement here. My point is still same. Both of them are circles. I mean spheres. Both of them are violet. But see, मैंने दोनों violet बनाए हैं. You can see. मैंने दोनों sphere बनाए हैं. But the arrangement is different. So first difference between them is their arrangement. Clusters are staph, chains are strap. Say yes. So the first difference is the arrangement. I want you to appreciate this. Can you see these are the chains? So this is Streptococcus. Streptococcus is in chain. Can you see these are in clusters? अंगूर के गुच्छे, the clusters of the grapes. है ना? Grapes के clusters के जैसा लगता है. यही word हम use करते हैं. The cluster of the grapes. These are known as Staphylococcus. Okay जी, है ना? Staph are in clusters, strap are in chain. The first thing हमें समझ में आई. ठीक है? ये तो चलो पहला difference है. Tell me one more difference between staph and strap. Staph, strap. This is staph. This is strap. The first thing we have seen the arrangement. These are in clusters. हमने देख लिया. ठीक है? And these are in chain. हमने ये भी देख लिया. But imagine I'm looking a slide. उस स्लाइड में आई कैन सी देयर इज अ वॉयलेट कलर कोकाई, वॉयलेट कलर स्फीयर, सो आई एम श्योर दैट इट इज अ जीपीसी, लेकिन आई एम नॉट श्योर कि वो क्लस्टर में है कि चेन में, कहीं पे क्लस्टर में दिख रहा है, कहीं पे चेन में दिख रहा है, सो आई एम गेटिंग कंफ्यूज्ड. So I'm sure that it is a GPC because it is violet color and it is a kokai. I'm sure it is one of them in the slide, but I'm unable to find out the arrangement very clearly. At some places I feel like it's a cluster. At some places I feel like it's a chain. What should I do? Is there is a confirmatory test? This is not confirmatory. This is vague. This is a vague criteria. But what is a confirmatory test? There is one confirmatory test which is known as catalase test. So you have to apply the catalase test. Staphylococcus are catalase positive and streptococcus are catalase negative. First learn the conclusion, then I will explain. ठीक है, confirmatory test is catalase, है ना. Screening test you can see is the arrangement, है ना. So confirmatory test is the catalase. See, this is catalase test. I will explain. पहले देखो तो सही. I will explain everything. देखो, can you see here? Can you see the two slide? यहाँ पे देखो bubbles आ रहे हैं. The bubbles are present. Presence of bubbles is catalase positive. So staphylococcus is catalase positive. And absence of bubble, no bubble is catalase negative. So this is streptococcus. I will explain कि bubble क्या है ना? No, not coagulase Saeed. Catalase. Coagulase will come later on. देखो धीरे धीरे चलो, जितना मैं बता रही हूँ उतने से समझते जाओ. ठीक है, let me finish the 2 chapters, then you ask your doubt. Here is no coagulase test. Catalase differentiates staph and strap. है ना. Coagulase बाद में आएगा to differentiate the staphylococcus further. ठीक है. So catalase differentiates staph and strap. My point is that learn the conclusion. पहले conclusion learn करो, then I will give you the detail. The test is catalase test, है ना. So there are two possibilities: catalase positive, catalase negative. So who is catalase positive? Say ma'am, staphylococcus is catalase positive because bubbles are coming, and say ma'am, streptococcus is catalase negative because no bubbles are coming. So you may be thinking, ma'am, ये bubble होता क्या है? Imagine. Okay, before that, see the diagram of staph and strap. This is staph, this is strap. I'm drawing 2 diagrams in front of you. This is the bacteria, this is the nucleus. This is the bacteria, this is the nucleus. In the cytoplasm, an enzyme is present. The name of that enzyme is catalase. Actually, catalase एक enzyme है जो cytoplasm में. So here it is present and here it is absent. This is the meaning of catalase positive, catalase negative. हम बोलते हैं ना कि भाई staph catalase positive है, strep catalase negative है. We say like this. So what do you mean by that? So it is an enzyme which is present here. That's why it is catalase positive. Here there is no enzyme such name. So this is catalase negative. This is the meaning. Catalase is an enzyme which is present in the cytoplasm here but not here. You got my point? Now how to test it? Imagine I'm having 2 specimens. This is my first specimen, this is my second specimen. है ना? I'm sure both of them contain GPC. I'm damn sure because they are wallet spheres. I'm sure. But I don't know which one contains staph, which one contains strap. I'm not sure. One of them is staph, one of them is strap. I want to know which is staph, which is strap. I'm sure that both of them are GPC. मेरा बात समझ में आई क्या? मेरी बात यह है कि दोनों में जीपीसी है, इसके लिए तो मैं डैम श्योर हूं 110%, क्योंकि मुझे इनके अंदर वॉलेट स्फीयर दिख रहे हैं, लेकिन मुझे नहीं पता किसमें स्टेप है किसमें स्ट्रैप है, मुझे वही तो पता करना है ना. सो आई विल टेक 2 slides. Got my point? I'm taking a slide here, and I'm applying the catalase test in both of them. So how to apply the catalase test? Take 1 drop of H2O2 in both the slide. H2O2 is hydrogen peroxide. Take hydrogen peroxide here also. It's a liquid. It's available in the lab. है ना? It's available commercially. 1 drop H2O2 in both the slides. समझना, please understand. Take 1 drop of your specimen here, mix it. Take 1 drop of your specimen, mix it. Now I don't know which is staph and which is strap. Imagine this one is staph. I don't know, but imagine, and this one is strap. Imagine, है ना? So if it is staph, in the cytoplasm of the staph, catalase is present. Say yes or no. So that catalase will degrade the H2O2. The catalase present in the cytoplasm of the staph will degrade H2O2 into H2O plus oxygen. So as soon as you mix the two, you can see the bubbles of the oxygen are coming on mixing the two. But here, whenever there is a streptococcus in the cytoplasm, there is no catalase, so H2O2 will not be degraded. It will remain as H2O2 even on mixing. So here no bubbles will come. समझ में आई क्या बात? So ये जो bubbles आते हैं ना, bubbles are of oxygen. Bubbles किस चीज के हैं? Oxygen के. और वो क्यों आते हैं? Because Staphylococcus have the enzyme catalase. Catalase degrade H2O2. Say yes, people, say yes. Just suppose I don't have a test tube here on my desk. Just suppose this is a test tube, है ना? In this test tube there is a specimen. Just suppose, है ना? I'm having GPC in that, I'm sure, but I don't know step or strap. So what I will do? I will take a slide. I will take 1 drop of H2O2 and I will put 1 drop of my specimen. I will mix with a stirrer and in 1 minute I will see the bubble. Bubble आया, step. Bubble नहीं आया, strap. That's as simple as that. See people, this is catalase test. Got my point? So you can see, you can get this image in your exam. It's an image based question. इस image पे 3 question. पहला question, identify the test. So looking at the image, you can identify bubbles वाला test is catalase. So answer will be catalase. है ना? Not coagulase, not MR, not VP. Answer क्या है? Catalase. Bubbles वाला test is catalase. पहला question, सबसे simple question, identify the test. दूसरा question, they will name it. This is bacteria A, this is bacteria B. So they will ask you who is A. So A is any bacteria which is catalase positive like Staphylococcus. B is any bacteria which is catalase negative, that is Streptococcus. So they can ask you to identify A and B. The third question, they can ask you the bubbles are of which gas? Is it oxygen, nitrogen, hydrogen, helium? कौन सी gas है? So it is oxygen. So I mean to say, मतलब concept एक ही है, but you can get many questions on that concept. समझ में आ रही है क्या बात, है ना? मैं सारे क्वेश्चन सॉल्व नहीं कराऊंगी भाई, बताती जाऊंगी यहाँ पे क्वेश्चन है, यहाँ पे क्वेश्चन है, यहाँ पे क्वेश्चन है. So you have to understand it, है ना? Mark in your notes whatever I have given. So can we proceed? Can we proceed, G, everyone? So this is catalase test, है ना? So this is catalase test. So we have seen, we have started GPC. Basically we have started GPC and we have seen we have 2 GPC in our syllabus. Staph and strap, है ना? This is staph and this is strap. We have seen 2 differences between them. Hannah, we have seen 2 differences. Number 1, this one is in cluster. Number 1, this one is in chain. Number 1 is arrangement. The arrangement is different. Number 2 difference. This one is catalase positive. This one is catalase negative. That is, here catalase enzyme is present, which is giving the bubbles. Here no catalase enzyme, no bubbles. Okay, till now we have learned that. This is the summary. This is the summary. Okay, Ji. Now let me start the first bacteria, staph. पहले मैं staph पूरा पढ़ाऊंगी. It will take आधा पौन घंटा लगेगा. It will take 40-45 minutes. It's a big bacteria. So don't rush. आराम आराम से पढ़ेंगे, है ना? और strap उससे भी बड़ा है, उसमें तो 1 घंटा भी लग सकता है. Very big, है ना? So these 2 bacteria are the biggest from all the bacteria. So we will give time to them. Rest of the bacteria is very small, but these 2 are big one. So we will give adequate time and understand it. ठीक है? The speed is good. You people are comfortable. It's not fast. Huh? It's average. You can understand. Comment. Is it good? Hannah, so let's start it. So let's start with Staphylococcus. I'm starting with chapter number 1 today. Okay? Yes, so let's start with Staphylococcus. Okay? So Staphylococcus. Now let's further classify. We have seen GPC, how to classify GPC. So whenever there is a word GPC, the first test you have to apply is catalase. हमेशा पहले apply करो catalase. Look which is the GPC. If the catalase positive or catalase negative. If it is catalase positive, it's staph. If it is catalase negative, it's strap. ये तो हमने देख लिया, बहुत बार देख लिया. Strap को छोड़ो फिलहाल. We will take it later on. Later on, chapter number 2. We will take it later on. Let's continue staph. Now, how to further classify staph? इसको आगे और कैसे classify करूं? In our syllabus, total 3 type of Staphylococcus are there. So how to classify them further? So the next test is coagulase. Coagulase. Someone was saying coagulase ना, तो coagulase यहाँ आएगा. मेरी बात सुनो, कान खोल के, है ना? Only one staph is coagulase positive. जो मेन हीरो है, staph फैमिली का मेन हीरो कौन है? Staph aureus. He is the main hero, है ना? And we have many side hero. तो side hero are coagulase negative. So coagulase positive is the main hero, and coagulase negative will be the side hero. तो side heroes का क्या नाम है? Who are side hero in this movie? हाँ? Who was side hero in this movie? So it is known as coagulase negative Staphylococcus cones. Side heroes को मिला के एक नाम दे दो, सारे side heroes को. They are known as cones. What is the full form of cones? Coagulase negative Staphylococcus. These all are coagulase negative. ठीक है? These all are coagulase negative. So see the same thing? See the same thing here? So first you apply catalase. Catalase will differentiate staph and strap. If it is catalase positive, it's staph. If it is catalase negative, it's strap. ये तो सबको समझ गया, है ना? The next test you apply is coagulase. Coagulase positive is main hero of the Staphylococcus. एक ही होता है main hero तो Staph aureus. बाकी सारे side hero. All of them are side hero. तो side hero are known as cones. है ना? Coagulase negative Staphylococcus. They all are coagulase negative. Now we have 2 side heroes in this movie. Total 3 heroes are there. One is main hero and 2 are side heroes. है ना? हमने movies देखी है ना बहुत सारी, जिसमें एक main hero होता है, 2 side hero होते हैं, है ना? So these 2 are side heroes, है ना? Just for comedy these are there. ठीक है? 2 side heroes are there. So name them. Say Staph epidermidis and Staph saprophyticus. We have 2 side heroes. How to differentiate these 2 among themselves? इन दोनों को भी तो differentiate करो. So the next test is novobiocin. What's the name of the test? नाम बोलो. Novobiocin. देखो novobiocin एक chemical है. What is novobiocin? Like penicillin, like cephalosporin. It's a drug. एक दवाई है, एक केमिकल है, इट्स अ ड्रग, है ना. सो एपिडर्माइटिस के ऊपर डालोगे तो एपिडर्माइटिस विल डाई, है ना, वो मर जाएगा. नोवोबासिन डालते से एपिडर्माइटिस विल बी किल्ड. सो इट इज ससेप्टेबल, इट इज ससेप्टेबल टू नोवोबासिन. लेकिन सैप्रोफिटिकस इज वेरी स्ट्रॉन्ग. आप नोवोबासिन डाल दो, इट विल नॉट किल. इट इज रेजिस्टेंट, इट विल नॉट किल. So saprophyticus is resistant. So put few drops of novobacin and see whether it is killed or not killed. If it is killed, I mean it is susceptible, it is epidermidis. If it is not killed, I mean it is resistant, it is saprophyticus. So there are 3 tests. People, concentrate on your screen. Catalase differentiate what and what. Say ma'am, staph and strap. Coagulase differentiate what and what. Say ma'am, staph aureus from others. And novobacin differentiate what and what. The two cones, epidermidis and saprophyticus. मेरी बात समझ में आई? Which test is differentiating what? So this is your questions, है ना? I said कि catalase test किन दो को differentiate करता है? You tell me. Coagulase test किन दो को differentiate करता है? और novobacin किन दो को differentiate करता है? हाँ? These all are your question. चलो. Catalase differentiate staph and strap. सबको पता है. Staph positive है, strap negative है. Coagulase differentiate staph aureus from other staph, that is cones. यहाँ positive है, यहाँ negative है ना? And novobacin differentiate the two side hero. स्टेप एपिडर्माइडिस, स्टेप सैप्रोफिटिकस, है ना? ये ससेप्टेबल है, ये रेजिस्टेंट है. यहाँ पॉजिटिव नेगेटिव नहीं बोलेंगे. समझ में आई क्या बात? कितनों को समझ में आई मेरी बात? हाउ मेनी ऑफ यू गॉट माय वर्ड्स? हाउ मेनी ऑफ यू गॉट दिस कॉन्सेप्ट? रिस्पॉन्ड पीपल. हाँ, इफ यू गॉट माय कॉन्सेप्ट ना, आंसर 7 क्वेश्चन. 7 क्वेश्चन पूछूंगी, 7 के आंसर देना है. पहले मुझे क्वेश्चन पूछने दो. फर्स्ट लेट मी आंसर 7 क्वेश्चन, देन यू स्टार्ट आंसरिंग. यू आंसर इन सीक्वेंस ओनली, ठीक है? So I will ask you 7 questions. 1, 2, 3, 4, 5, 6, 7. One by one. ठीक है? My first question, enumerate GPC. My second question, tell me a GPC which is catalase negative. ठीक है? My third question, tell me a GPC which is catalase positive. I'm sorry, yeah, I'm right. ठीक है? Tell me a GPC which is catalase positive and coagulase also positive. ठीक है? Tell me a GPC which is catalase positive but coagulase negative. ठीक है? Tell me a GPC which is catalase positive, coagulase negative, but novobacin susceptible. And last you got it, GPC catalase positive, coagulase negative, novobacin resistant. चलो, 7 question, 7 answer. कौन बताएगा? Who will tell? If it was a live class, one by one I ask you, you tell. ठीक है, but I can't see you all. I can see the chat box only. चलो, start answering. GPC कितने पता है? You will say, GPC are staph and strap. I cannot say staph or strap. I will say both. ठीक है, GPC is staph and strap. ठीक है, GPC which is catalase negative. It is negative, so it is strap. Which strap? I don't know. Streptococcus. GPC which is catalase positive. I will say staph. Which staph? I don't know. I will say just staph. है ना? GPC which is catalase positive, मतलब staph तो है, लेकिन coagulase positive तो एक ही staph होता है. Only one staph is coagulase positive. Answer is staph aureus. Start seeing the answer. Staph aureus. Yes. Now GPC which is catalase positive but coagulase negative. The remaining all the side heroes. The side heroes are cones. Which cones I cannot say. The full form of cones is coagulase negative staphylococcus. है ना? And the two they are asking novobacin susceptible, novobacin resistant. The novobacin susceptible जो बहुत डरपोक है वो है epidermidis, और जो बहुत पावरफुल है, ब्रेव है बहुत, वो है saprophyticus. ये वाला novobacin से मर जाता है और ये novobacin से resist करता है. ठीक है? I tried my best, है ना? So these all are your PYQs in different format. Language different होगा, format different होगा, लेकिन concept यही है. तो ये सब तुम्हारे PYQ है ना, या फिर approach based question. Okay, appreciate my efforts. समझ में आ रही है क्या बातें? हाँ? So this is the thing. So we have started. We have seen the classification. Can I remove this page? So I have written nothing additional. It is given in the classification. उसी को मैंने question format में कर दिया. Okay, ठीक है? So this is the classification we have learned. So same thing is given to you here also. You can see catalase positive, catalase negative. So you can see, ma'am, catalase positive is staphylococcus. I can see the bubbles. And here catalase negative is streptococcus. Here we can't see the bubble. ठीक है, उसके बाद आ जाओ coagulase पे. Coagulase positive is only main hero, staph aureus, और coagulase negative is cones. उसके बाद आ जाओ novobacin पे. Novobacin से मर गया, sensitive है, susceptible है तो epidermidis, और resistant है तो saprophyticus. I don't think anyone have. Yes, Arshad, absolutely right. You have given the answers in the sequence. Very good. So absolutely right. Appreciated your efforts. So let's move ahead. Okay. So done. Let's move ahead. So let me teach you the 3 staphylococcus one by one. So I'm teaching you the chapter Staph. ठीक है, my chapter number 1. I will teach you one main hero, Staph aureus, and two side heroes, Staph epidermidis and saprophyticus. One by one I will teach you all Staph, है ना? First I will teach you Staph aureus in detail. ये थोड़ा डिटेल में देखेंगे, मेन हीरो है, इनके बारे में वन वन लाइन, जरा जरा सा एक लाइन खत्म. So these are the side heroes. In this way we will pack chapter number 1 Staph. So can I start the main hero? Are you people with me? Can I start Staph aureus? Let's start Staph aureus, है ना? So Staph aureus का कल्चर मीडिया बताओ. आई एम टीचिंग यू Staph aureus, द मेन हीरो, कल्चर मीडिया. सबसे पहले टेक Staph aureus, है ना? देयर आर 3. सो Staph aureus एंड कोन्स. कोन्स में वी हैव Staph epidermidis and Staph saprophyticus. Understand one more difference between them. Understand one more difference. This is pigment producing. It form a pigment. They do not form a pigment. Which color pigment it form? Golden yellow color. इसकी colonies are also golden yellow color. The colonies here no pigment, so the colonies are white. The colonies are white, है ना? इसलिए इन दोनों को मिला के Staph albus भी कहते हैं. albus यानी white colony. तो ये दोनों white colony बनाते हैं, no pigment, है ना? So I'm teaching you Staph aureus, the main hero. As I told you, it produce pigment. Pigment produce करने वाले दो ही बैक्टीरिया हैं. The one currently I am teaching you, Staph aureus. एक और पढ़ाऊंगी, आज ही पढ़ाऊंगी. Today only I will teach you Pseudomonas. है ना? So this produces golden yellow pigment, है ना? And this will produce many pigment, but most important among them is green. Green भी करता है, blue भी करता है, red भी करता है और yellow भी करता है. But most common is green. So this one is golden yellow. So pigment producing two bacteria in your syllabus. ठीक है? The first we are studying right now. Okay? So Staph aureus. So Staph aureus on nutrient agar, it is known as oil paint appearance. Or it is known as oil paint. ऐसा लग रहा है yellow color का oil paint के drops गिर गए. It's looking like yellow color oil paint drops are there. It is known as oil paint appearance. It's a PYQ asked 100 times. Oil paint appearance on nutrient agar. It's Staph aureus. What is the reason for oil paint? Say ma'am, pigment. Which color pigment? Yellow, golden yellow pigment, which is produced by Staph aureus. That's why the color of the colony is golden yellow, and that's why it's looking like a oil paint. Oil paint का drop गिर गया है. So it is looking like oil paint appearance. Say yes on the nutrient agar, है ना? On blood agar it is showing beta hemolysis. चलो एक बार मैं कहानी खत्म करूंगी, है ना? ब्लड अगार सिर्फ एक बार पढ़ाऊंगी. हर बैक्टीरिया में मैं तुम्हें ब्लड अगार पढ़ाऊंगी. इन एवरी बैक्टीरिया आई विल टीच यू ब्लड अगार, है ना? ब्लड अगार में 3 पॉसिबिलिटीज हैं, अल्फा हेमोलिसिस, बीटा हेमोलिसिस, गामा. वंस फॉर ऑल आई विल टीच यू ऑल. बार बार नहीं बताऊंगी, ठीक है? सो प्लीज अंडरस्टैंड. आई एम हैविंग 3 ब्लड अगार. सी, दिस इज अ ब्लड अगार, दिस इज अ ब्लड अगार, दिस इज अ ब्लड अगार. प्लीज अंडरस्टैंड द मीनिंग ऑफ अल्फा बीटा गामा. ठीक है? रटने के लिए तो रट लोगे, बट प्लीज अंडरस्टैंड. सो दिस इज द 3 ब्लड अगार. Here is the colony of a bacteria. Here is the colony of a bacteria. And here is the colony of a bacteria. This is a bacterial colony I have drawn. On blood agar, we have blood. I'm sorry. On blood agar, we have blood. Now we have RBCs. So these all are RBCs around the colony. हर तरफ है RBCs. Everywhere we have RBCs. I'm not drawing everywhere. I'm just drawing around the colonies. है ना? RBCs are everywhere. ठीक है? Here also we have the RBCs. RBCs are everywhere because it is a blood agar. We have RBCs everywhere. Now there are 3 possibilities. Please understand. Anna, the first colony, the bacteria will hemolyze the RBC. It causes rupture of the RBC around the colony, but it causes partial hemolysis. It causes partial hemolysis. Partial hemolysis of RBC. Partially it is degrading the RBCs around it. So around the colony you will get a green color zone. It is known as alpha hemolysis. Around the colony. Colony तो है ही है. Around the colony you will get a green color zone. It is known as alpha hemolysis. Alpha hemolysis is partial hemolysis. Some bacteria degrade the RBC partially. There are some bacteria which degrade the RBC completely. They degrade the RBC completely. Complete hemolysis is known as beta hemolysis. Beta is complete. है ना? So around the colony, you will get a transparent zone. है ना? I don't have transparent color. I'm using yellow, but actually it's a transparent zone. Here it's a green zone. Here it's a transparent zone. ठीक है? And the third possibility, the bacteria is not at all hemolyzing the RBC. Not partial, not complete. So say ma'am, no hemolysis. No hemolysis is gamma. No. So alpha is partial, beta is complete, gamma is no. So around the colony, here you get green color zone, here you get transparent zone, and here you get only colony, no zone. तुम्हें यहाँ पे सिर्फ colony मिलेगा. Colony के around you don't get anything. So say people say, so for every bacteria, either the bacteria is alpha or beta or gamma hemolytic. और क्या possibility है ना? Alpha means partial hemolysis, beta means complete hemolysis, and gamma means no hemolysis. Hemolysis किस चीज का? RBC का. Got my point? So here you get around the colony, you get a green color zone. You get a green color zone. Here around the colony, you get a translucent transparent, translucent zone. And here around the colony, you get no zone, only colony. How many of you got it? So please understand, this is the basic. ये तो basic है, मतलब this is the KG level I am teaching you. भाई मैं assume कर रही हूँ कि ये तो तुम्हें आता ही है अल्फा बीटा गामा. So अब हर बैक्टीरिया में मैं जितनी भी बैक्टीरिया पढ़ाऊंगी ना आज 25 सब में मैं बोलूंगी either अल्फा और बीटा और गामा. You have to learn alpha, beta, gamma. Now you understand the meaning. So for step or years, learn the word beta. You know what is beta? It's complete. So I will say beta only. है ना? So it's beta. So you can see अगर zoom करके देख पाओ तो you can see the colony और around the हर colony के around एक translucent सा zone है. इतना अच्छा दिख नहीं रहा. The image is not very clear. But if you can appreciate, you can imagine actually around the colony there is a translucent zone. It's beta. It's beta. So on nutrient agar say ma'am oil paint. On blood agar say ma'am beta. है ना? On PPA. I don't remember the full form of PPA. It's a chemical. On PPA agar, the pink color colonies are there. You can see the pink color colonies on passing the gas ammonia. Not very important. The last one is selective. Selective. Achha, what is selective media? Yesterday I taught you selective. Enrichment media and selective. कौन बताएगा selective क्या है? Yesterday I taught you. Enrichment media भी बताया मैंने, selective भी बताया. What is selective media, people? कल बताया था ना selective media. ऐसा media, it is a media in which we are adding either stimulatory substances for the bacteria of choice or inhibitory substances for the bacteria which we do not want to grow, and it's solid. Very good. It's solid. So yesterday have I taught you कि तुम्हें 25 की bacteria की selective media याद करने हैं? Have I taught you this? Have I told you this? Have I given you a table also yesterday in the general? हाँ, जिसमें पूरे 25 bacteria are enumerated. I told you today, after today's session, you read that table. बोला था क्या मैंने? हाँ, तो हर बैक्टीरिया का मैं सिलेक्टिव बताऊंगी. If you wish, उस टेबल में चेक करते जाओ. If it is not there, you can add. ठीक है? You have to learn the selective media of all. So selective media are 3 here. कितने हैं? 3 हैं. 3 हैं. There are 3. The first is the ludlam. You will say, मैम ये ludlam क्या होता है? मतलब क्या? Ludlam तो name of scientist है, भाई जिसने उसको बनाया है. तो ludlam में न्यूट्रिएंट अगार ले लो. न्यूट्रिएंट अगार को मैं वेज थाली बोलती हूँ. वेज थाली फॉर द बैक्टीरिया. न्यूट्रिएंट अगार पे सब कोई ग्रो करता है. दुनिया का हर बैक्टीरिया ग्रो ऑन द न्यूट्रिएंट अगार एक्सेप्ट फ्यू. है ना? वेज थाली तो सब कोई खाता है. लाइक दैट. है ना? सो दिस इज न्यूट्रिएंट अगार. न्यूट्रिएंट अगार में तो सब कोई ग्रो करेगा. बट यू एड टेल्यूरियम क्लोराइड, लिथियम क्लोराइड एंड टेल्यूरियम in the nutrient agar. अब कोई भी ग्रो नहीं करेगा. Only step will grow. So you take nutrient agar. This is nutrient agar and add 2 things: lithium chloride and tellurium. है ना? So no other bacteria will grow. Only step aureus will grow. So it becomes selective for step aureus. समझ में आई क्या बात? समझ में आई? So this is ludlam, number 1. Number 2, mannitol salt. Again nutrient agar में 2 चीजें मिलाओ, mannitol and salt. So it becomes selective for step aureus. Only step aureus will grow, others will not grow. And third is Columbia. कोलंबिया में बना होगा. इट्स अ प्लेस. है ना? कोलंबिया में बना होगा. इसमें हमने 2 चीजें डाली, कोलिस्टिन एंड नैलेटिक एसिड. डोंट वरी. लिसन. सिलेक्टिव मीडिया. लर्न द नेम्स, डोंट लर्न द कंपोजिशन. ठीक है? द फर्स्ट इज लुडलाम, नेम ऑफ द साइंटिस्ट. है ना? सेकंड इज मैनिटोल सॉल्ट, इट्स अ कंपोजिशन. है ना? एंड थर्ड इज कोलंबिया. नाउ इफ यू विश यू कैन लर्न द कंपोजिशन आल्सो. इट्स योर चॉइस. पूछा तो नहीं है अभी तक. इट्स नॉट अ पीवाईक्यू, लेकिन दे कैन आस्क. है ना? सो दे ऑल आर न्यूट्रिएंट अगार ओनली. न्यूट्रिएंट अगार तो है ही. एडिशनल क्या चीज है? लुडलाम में एडिशनल है लिथियम एंड टेल्यूरियम. मैनिटोल सॉल्ट में नाम में ही है मैनिटोल एंड सॉल्ट. और कोलंबिया में है कोलिस्टिन एंड नैलेटिक एसिड. इफ यू वांट टू लर्न, इट्स गुड. इफ यू डोंट वांट टू लर्न द नेम्स. यू कांट मिस द नेम. नाम तो याद करो कम से कम. नेम्स पे पीवाईक्यू है ना? कंपोजिशन पे अभी तक तो नहीं है, बट आई कैन नॉट टेक द गारंटी. ठीक है? पूछने को तो कुछ भी पूछ सकते हैं. ठीक है? गॉट माय पॉइंट? है ना? हाँ बोलते जाओ. सो प्लीज लर्न द 4th थिंग. स्टेप ऑरियस पढ़ा रही हूं मैं, पहला बैक्टीरिया आज का. है ना? मुझे 4 चीजें बताओ. न्यूट्रिएंट अगार पे क्या अपीयरेंस होगा? ब्लड अगार पे क्या अपीयरेंस होगा? पीपीए अगार पे क्या अपीयरेंस होगा? और सिलेक्टिव मीडिया इन्यूमरेट करो. 1 2 3. चलो, से द आंसर्स. क्वेश्चंस आर इन फ्रंट ऑफ यू. दीज ऑल आर योर पीवाईक्यूज. चलो, न्यूट्रिएंट अगार पे से 1 वर्ड. से मैम ऑयल पेंट अपीयरेंस. द रीजन फॉर ऑयल पेंट इज गोल्डन येलो पिगमेंट. ब्लड अगार पे से मैम बीटा. इट्स बीटा, कंप्लीट हेमोलिसिस. है ना? बीटा. ठीक है? पीपीए पे से मैम पिंक कॉलोनी. पिंक कॉलोनी, यू हैव टू लर्न पिंक कॉलोनी. सिलेक्टिव तो लर्न करने ही पड़ेंगे. नहीं करोगे आई विल स्लैप यू ऑल. यू हैव टू लर्न सिलेक्टिव. सिलेक्टिव भूल नहीं सकते. इफ यू आर मिसिंग द सिलेक्टिव, यू आर आउट ऑफ कंपटीशन. है ना? यू आर मिसिंग 1 क्वेश्चन आउट ऑफ 300. 1 गारंटेड क्वेश्चन आउट ऑफ 300. यू कैन नॉट डू सो. इतने बड़े फूल तो नहीं होगे कि गारंटेड क्वेश्चन छोड़ के जाओगे. है ना? सो डोंट मिस इट. ओके, सो लिसन. What I am saying, the 3. So please enumerate. भूल गई मैं क्या था. It's ludlam. ठीक है? The second is the mannitol salt. The third is the Columbia. Please learn that. You know the compositions also. So we are done with the culture media. Okay, we are done with culture media. Now after culture media, we are coming on the virulent factor. Virulence, factor of virulence. Virulence के दो factor होते हैं यहाँ पे, toxins and enzymes. So first I will teach you the toxins, then I will teach you the enzymes which are secreted by Staph aureus. Staph aureus have toxins, 4 types of toxins, and Staph aureus have enzymes, 2 type of enzymes. One by one we will see. So let's come on the toxins of the Staph aureus. Staph aureus की toxins की बात करो. There are 4 toxins it is secreting. Let me tell you the names. है ना? उसके नाम में ही उसका meaning है. आधा माइक्रोबायोलॉजी तो उसके नाम में ही हो जाता है. ठीक है? The first is cytolytic toxin. The second is enterotoxin, toxic shock syndrome toxin, epidermolytic toxin. Let me explain. ठीक है? Listen. Cytolytic, split the term. Cyto मतलब cell, lytic मतलब degradation. Cytolytic, it is degrading the cell. So it will produce pus everywhere. ये वाला toxin मेरी body में, just imagine in my body this Staph aureus is entering. ठीक है? In my body this Staph aureus is entering. So cytolytic, it will secrete all 4 toxin in my blood. ठीक है? So cytolytic toxin will degrade my cells, human cell, cytolytic, and it will produce pus everywhere. Entero मतलब क्या होता है? Intestine. Enterocyte सुना है? Enterocyte is a cell of the intestine. ठीक है? Okay. Enterotoxin, it will act in the intestine and it will cause food poisoning. ठीक है? The third is the toxic shock syndrome toxin, TSST. It will cause toxic shock syndrome. And last is epidermolytic toxin, epidermis of the skin, epidermis, dermis. It will cause a disease, SSSS. Listen, I'm going to teach you all these 4 diseases one by one. Here I'm not teaching you the diseases. So don't concentrate ki ma'am which is causing which disease. Concentrate on the name of the toxin. There are 4 toxins I'm teaching you. Which 4 toxins? Cytolytic, entero, toxic shock syndrome, and epidermolytic. They all will cause one. Total 4 diseases I'm going to teach you right now. Each of them caused by one toxin. Say yes. Say yes. Okay. 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 So I will not use Hindi. I will take care of it. Hardly I'm using Hindi. Yaar. Come on. I'm not using it. Okay. So I will take care. Still I will take care. Okay, ji. I will concise in the simplest form of English. Will it work? Okay. I'm sorry if I forgot. Many students don't understand Hindi here. Okay, ji. Listen now. So out of them, these 3 are super antigen. Yesterday I taught you super antigen. Say yes. Yesterday I taught you super antigen. Have I taught you 10 examples of super antigen yesterday? बताए थे क्या कल? Yesterday I taught you the 5 bacteria, the 4 virus and 1 fungus. बताए थे? हाँ, have I taught you? In the 5 bacteria, one of the bacteria was Staphylococcus. I taught you these 3 toxins of the Staphylococcus are super antigen but not cytolytic. So these 3 are super antigen. You can cross check if you wish. You can open and cross check these 3 toxins. So the toxins are done. Can you please enumerate and help me? Toxins. How many toxins? Please tell me the toxins of the Staph aureus. Tell me the 4 toxins. 1, 2, 3, 4. 4 toxins of the Staph aureus. The first toxin is cytolytic. The second toxin is the enterotoxin for the food poisoning. Ye pus karayega. This will cause pus. This will cause food poisoning. The third one is the toxic shock syndrome toxin. It will cause toxic shock syndrome. And the last one is the epidermolytic. Epidermolytic toxin. It will cause SSSS. Out of them, I'm saying these 3 are super antigen. Just learn that. These 3 are super antigen. Okay, this is all about toxins. After the toxins, let's come on the pathogenesis. The pathogenesis. I'm sorry. Enzymes you can skip. Okay, ठीक है. Come on the pathogenesis. Now tell the diseases. I told you 4 diseases. Na? I told you. Cytolytic causes pus. Pus everywhere. This is pus. It is caused by cytolytic toxin. Food poisoning is caused by enterotoxin. Toxic shock syndrome is caused by toxic shock syndrome toxin. And SSSS, scalded skin syndrome, Staphylococcus scalded skin syndrome, it is caused by epidermolytic toxin. So the 4 toxin causing the 4 diseases. There are 4 diseases. First, the cutaneous and deep pus, everywhere pus. In human body, whatever pus you know, pus, what forms of pus you know, maybe cellulitis, maybe abscess, maybe furuncle, maybe folliculitis, any type of pus in human body, the most common cause is Staph aureus. Do you have pus-filled lesion anywhere on the body? The most common cause is Staph aureus. Pus everywhere. Anywhere in the human body. Number 1. ठीक है? Number 2, food poisoning. Whenever food poisoning question come in your exam, you have to see 3 things. Not only this, I'm going to teach you many food poisoning today, tomorrow, a day after tomorrow. There are many organisms which cause food poisoning. Staph aureus is the first I'm teaching you. But I will teach you food poisoning is a syndrome. So first thing you have to see the food. What food the person is eating after which the person is having food poisoning. Different organisms have different affinity. For example, currently I'm teaching you Staph aureus. Staph aureus occurs after eating dairy product. You know what is dairy product? The cheese, paneer, ghee, है ना, milk, the dairy products. ठीक है, dairy products or meat, not meat, fish. Dairy product or fish, है ना, fish or meat. ठीक है ना. Step aureus. Bacillus cereus I will teach you today only. It occurs because of Chinese rice, Chinese fried rice to be specific. ठीक है. I will teach you one more today, Clostridium botulinum. It occurs because of canned food, है ना. Canned जो खाते हैं ना, cane में होता है food, canned food. So different bacteria, different organism have different food affinity. You have to learn this list. I will provide you this list also. You just keep on understanding. Everything is written with you. You get the concepts, I mean to say. So first thing, you have to see the food. Second thing, you have to see, is it diarrhea or vomiting or both? है ना. Some organism causes only diarrhea and food poisoning. Some causes prominently vomiting and some causes both. Let's talk about the Staph aureus. It causes both. It causes both. Currently, I am teaching you Staph aureus, but I am teaching you overall approach. You are getting my point. And the third thing, you have to see the incubation period. I mean, after eating, after how many hours the patient presents with the symptom. There are three possibilities. If it is within 2 to 6 hours after eating, 6 to 16 hours after eating, or more than 16 hours. So there is a list. There is a different list. You will have a differential diagnosis there. समझ में आ रही है? I will teach you food poisoning tomorrow, the complete syndrome. Here I am giving you just approach. So currently Staph aureus come in this zone. So Staph aureus, let me concise myself up to Staph aureus only. है ना? Staph aureus occurs due to dairy product and fish. It occurs within 2 to 6 hours and both diarrhea and vomiting occur. This is for Staph aureus. So different organism, the different combinations are there. समझ में आई क्या बात? So whenever in your exam you get a question on food poisoning, a big question on food poisoning, you have to notice three things. It will be a big question, but everything, the rest information is useless. Question में से you have to pick three information. बड़े से big question में से, big clinical question, you have to pick up three things. Number one, try to find out the food. If they mention the food in the question, after eating what the patient have the symptom. Try to find out, figure out whether the patient have only diarrhea, only vomiting, or both. And try to figure out the incubation period. If you can do so from your question, you can come on the conclusion. Which type of food poisoning is it? ठीक है. So I have given you an overall approach, but currently I am teaching you food poisoning due to Staph aureus. My topic is Staph aureus right now. Okay. So here incubation period is 2 to 6 hours. It occurs because of eating of the dairy product and fish and meat. है ना? And here diarrhea, vomiting both occur. So let me tell you the mechanism of both. See beautiful diagram. Everyone see, this is milk. Say yes. है ना? This is pizza containing the cheese. है ना? Dairy product. है ना? This is fish and this is meat. See the diagram having all the food here mentioned here. So all the food. है ना? So it contains the bacteria. See the bacteria in clusters. Beautiful diagram. See the color, see the shape, see the cluster. तीनों चीज appreciate करो. I want you to appreciate the violet color. I want you to appreciate their sphere. I want you to appreciate they are in cluster. So looking at the diagram itself, it's a spotter. You can identify, man, Staph aureus. It's Staph aureus, no other bacteria. It's Staph aureus. है ना? It is producing a toxin. The person is eating the toxin, not the bacteria. It is a preformed toxin. It is a preformed. The toxin is formed in the food itself. The person is not eating the bacteria. Mind my words. The person is eating the toxin secreted by the bacteria and the food. The food contain the toxin. Food do not contain the food contain bacteria, but bacteria secrete the toxin and die. Now there is no bacteria. It contains toxin. The toxin go inside. Which toxin out of the four? Out of the four, which toxin? Name it. Say, ma'am, enterotoxin. It's not toxic shock syndrome. It's not cytolytic. It's not epidermolytic. It's enterotoxin. Enterotoxin do two things. It will do two things. Number one, it will go to intestine and causes peristalsis. Causes peristalsis that leads to diarrhea. ठीक है. Number two, it will go to the blood. Via blood, it will go to the brain. In the brain, it will go to vomiting center that causes vomiting. So person have diarrhea and vomiting both. ठीक है. So the mechanism of diarrhea is peristalsis and the mechanism of vomiting is vomiting center in the brain, stimulation of that. People say yes. है ना? I'm summarizing. Staph aureus causes 4 diseases. 1, 2, 3, 4. है ना? One I already taught you. It causes pus everywhere. Pus-filled pyogenic lesions. ठीक है? The reason for the pus is cytolytic toxin that we have ended. है ना? It can be folliculitis, furuncle, abscess, है ना? XYZ, many diseases. Second, the name of the disease I am teaching you right now is food poisoning. First, tell me the name of the toxin. Pehla question, name of the toxin. You will say, ma'am, it's enterotoxin. ठीक है? It's enterotoxin. ठीक है? I got it. It's enterotoxin. Very good. Now you tell me 3 things. Number 1, the food ka preference. Number 2, the incubation period. And number 3, is it diarrhea or vomiting or both? है ना? And tell me the mechanism also. ठीक है? So you answer one by one. ठीक है? You say, ma'am, food. Regarding food, it's dairy product. Dairy product it's fish, and it's meat. ठीक है, number 1. Incubation period, please learn by heart, it's 2 to 6 hours. ठीक है, it causes diarrhea also, vomiting also. है ना? The mechanism of diarrhea is peristalsis. ठीक है, and the mechanism of vomiting is vomiting center in the brain. That's it. No other question will come from here. The summary is crystal clear. Give me a thumbs up. Give me a thumbs up. Am I fast or you are getting it? Am I fast? Maybe my speed of saying is fast, but the concepts we are going slow. I'm a fast speaker. Okay, but concepts you are getting, na? It's not fast, is it? Okay, we can go with the same speed. So you got it. You get many questions on food poisoning. The summary is in front of you. Learn it by heart. Coming on the third disease. The third disease is toxic shock syndrome. The name of the disease is toxic shock syndrome. Name the toxin. Here the toxin is cytolytic. Here the toxin is enterotoxin. So similarly, here the toxin is toxic shock syndrome toxin. Okay, name of the toxin. So describe it now. Name the symptoms. Describe everything regarding toxic shock syndrome. What is toxic shock syndrome? I'm sure many people even don't know what is toxic shock syndrome. Okay, I will tell. Don't worry. So you see, toxic shock syndrome here occurs only in females. Only in females. In menstruating female. है ना? During menstruation, female uses tampons. She uses tampons. है ना? Sometimes the tampons get contaminated with TSST, toxic shock syndrome toxin. And the contaminated tampon, the toxin get entered in female body via vagina. Here the toxin was entering via mouth, enterotoxin. But toxic shock syndrome toxin enters via vagina through contaminated tampon, in a menstruating female. The biggest clue you get in the question is menstruating female. The question will start, there is a menstruating female. ऐसा ही start होगा question. Please learn. So always think of the differential this, है ना? Using tampons. So through tampons, the toxin is going inside. After going in blood, toxin going in multiple organ and causing multiple organ failure. It's not one organ, it's multiple organ. So the female will have fever, hypotension, shock, diarrhea, vomiting, rash. It can be anything. It's multi-system. Say yes, this is toxic shock syndrome. And the last one is the name of the disease is SSSS. What is the full form of SSSS? Staphylococcal scalded skin syndrome. Staphylococcal scalded skin syndrome, SSSS. The name of the toxin is epidermolytic toxin. Epidermolytic toxin, the last toxin, also known as exfoliative toxin. Here the epidermis gets separate from the dermis. Can you see? Epidermis and dermis got separated. It is known as Nichols sign. It's an image-based question. Nichols sign. I'm summarizing. I am done. I'm done. I'm summarizing. So you tell me Steph aureus. You will tell me. First, you tell me the name of the 4 toxin. 1, 2, 3, and 4. And the corresponding disease and the corresponding symptom. पहले toxin बताओ, फिर disease बताओ, फिर symptom बताओ. Say the toxin. Start saying the toxin. What are the 4 toxin? Say, ma'am, cytolytic toxin. Say enterotoxin. है ना? Say toxic shock syndrome toxin. And say epidermolytic, also known as exfoliative toxin. First, say the names. ठीक है जी? Now say the disease caused by them. Cytolytic causes what? This causes what? This causes what? Tell the disease. So here the name of the disease is pus. And very simple. Here the name of the disease is food poisoning. Here the name of the disease is toxic shock syndrome. And here the name of the disease is SSSS. Now can you describe something about them? Some symptoms and main points. So in the pus, it is multiple pus, folliculitis, furuncle, abscess, multiple pus filled lesion in the body. Enterotoxin I have described. Incubation period is 2 to 6 hours. I have told you the preferable food. It's dairy and fish. And it's diarrhea and vomiting both. And you know the mechanism of both of them. ठीक है? Toxic shock syndrome, it is through menstruating female. Write down. Enters through tampon. Contaminated tampon through vagina and multi-organ failure. It's multi-organ failure. This is the summary. Last, SSSS. In SSSS, epidermis separate from dermis. They separate from each other. And this is known as Nichols sign. Nichols sign. You can see. It's Nichols sign. Okay, Ji, we are done. We are done. Okay, done. Give me a thumbs up. Can I flip this page? Can I flip this page? You can solve all the questions. So this is the summary. We are done. Okay? So we are done with the main hero, Steph aureus. In the Steph aureus, I told you 3 things. Just learn 3 things. The culture media, nutrient agar, blood agar, PPA agar, and selective media. Okay? The toxins, enumerate 1, 2, 3, 4 toxin. And the diseases, I mean the pathogenesis, 1, 2, 3, 4. Pack. The topic is done. Drug of choice, you can read by yourself as given in the chart. I will not read the drug of choice, and rest you will read in the pharma, the doses and all. Okay, I'm coming on the two side hero. What are the two side heroes we are having? The first side hero, just a second. The first side hero is Staph epidermidis, and the second side hero is Staph saprophyticus. Staph epidermidis and Staph saprophyticus, the two side heroes. You know about the two side heroes? Both of them are cones. है ना, coagulase negative staphylococcus. This one is novobacin sensitive, है ना, बहुत डरपोक है. It's very cowardly, you can say. And this one is very brave. है ना, this do not kill with novobacin. This one is killed with novobacin. Learn one disease caused by them. Staph epidermidis causes prosthetic wall endocarditis, the name of the disease. Prosthetic wall endocarditis in the heart. So the heart is involved. And this one causes UTI. So here urinary bladder, the urinary bladder and the urinary system. UTI and sexually active. So learn the organ. It is causing a disease in the heart, endocarditis. This one causes UTI in sexually active female. We are done. We are done with the chapter. Can we solve some questions? So we are done with the chapter Staph. Staph. Here I taught you three Staph. The one is Staph aureus and two cones. I taught you Staph epidermidis, Staph saprophyticus. Here I taught you many things. I taught you culture media. I taught you the toxins. I taught you the pathogenesis. Here I taught you one disease. That's it. Say something, people. Respond. This is a way to study the entire micro. It's very interesting subject. It's very interesting subject. Hana, subject is not boring or interesting. It's the person who is teaching you is boring or interesting. So am I boring or interesting? How do you find? Treatment, you read the drug of choice. It's given in the table. No? There is nothing to explain. I provided you these tables. I guess everyone have the printout. If you don't have printout, it's okay. The soft copy. See the last column. See the last column is a treatment. Learn drug of choice, drug of choice, drug of choice. That's it. That's it. Nothing to explain here. So learn the drug of choice. Doses will be dealt by the pharmacology. Okay? Chalo. Let's continue. So we will solve some questions on chapter number 1 and we will move on the next bacteria, chapter number 2. Okay, read the question, tell me the answer. Thank you for your compliment. Okay, read it. Tell me the answer. Tell me a bacteria which is catalase positive and novobacin resistant. Tell me a bacteria who is catalase positive and novobacin resistant. What is the answer here? What is the answer? What is the correct answer? Is it Staph aureus? Is it Staph epidermidis or saprophyticus? Who is novobacin resistant? Resistant se mujhe yaad aata hai very brave. Who is not killed with novobacin? Very brave bacteria. It's saprophyticus. So correct answer is saprophyticus. You all are right. I'm changing the question. Instead of resistant, I'm saying susceptible. Susceptible se mujhe yaad aata hai very cowardly. Very cowardly, I mean. बहुत ही डरपोक है, I mean to say. So what is the correct answer in that case? In that case, the correct answer will be epidermidis. Epidermidis. So you all can understand. Now the next question, oil paint appearance on nutrient agar. The easiest question. The easiest ever. The most easy question in the world. Oil paint appearance. Of course, the answer is Staph aureus. I'm not halting here. So of course, everyone knows the answer is Staph aureus. The reason for the oil paint is pigment production. The golden yellow pigment production. So you have to learn that. Now it's a clinical question. Read the question with me. Come on the conclusion. Take the clues. हमेशा clinical question में try to take the clues. ठीक है? So there is a 25-year-old female presented on the hospital on day 3 of menstruation. She is a menstruating female. Menstruating female, the biggest clue. Menstruating female, day 3 of menstruation, she is complaining. She is having fever. She is having vomiting. She is having rash. 3 complaints. What is the diagnosis? So menstruating female से ही तुम्हारा diagnosis बनेगा toxic shock syndrome, having multiple organ failure. So correct answer is toxic shock syndrome. Any confusion? We are done with Staph. So questions का number I have reduced because we have to cover all the bacteria today. Na? If I keep on, solving many questions here, so the text will be left. In the end, theory will be left. Few bacteria will be left untouched. I do not want that. So question you do by yourself. Few prototype we are doing here. 2-3 question with each bacteria. But I suggest you to solve your question bank of entire bacteriology today only after the class. Okay? Can I move that to the next bacteria? The next bacteria is Streptococcus. So chapter number 2, bacteria number 2, Streptococcus. Can I start? You people with me? Let's start Streptococcus. Now let me warn. It's a warning. The warning is that Streptococcus is difficult. I'm not like scaring you. It's a real warning. It's little bit difficult. Especially the classification is difficult. You know the classification of Staphylococcus, no? Now let me tell you the classification of Streptococcus. It's little bit difficult, not much. I will try my best to explain you. So let me start with the classification. Whenever we start any bacteria, we see the classification and then we move on the details. ठीक है? In Staphylococcus, I taught you 3, 1 main and 2 side, है ना? In Streptococcus, I will teach you 5. Again, 1 main and 4 side, or 2 main and 3 side, whatever you say. So I will teach you 5 there. It's a big chapter, rather the biggest chapter. Okay? No, Medico, I will not teach you the master charts. I guess everything is understood. If you have any doubt, you can ask me here only. Bacteria by bacteria, you can read it. Like Medico is asking that shall I explain the master charts also? So I suggest you, I have completed step warriors now. Do you all have master chart with you? Say yes or no. First tell me, do you all have these charts with you? The master charts? Do you have either soft copy, hard copy? It's your choice. Do you have it or you don't have it? Whatever, whosoever have it, please open it right now. Hana? Hard, soft, whatever, open it. Now I have completed first bacteria step warriors now. I've given only main hero. I have not given the side hero. I want you to read it completely. I'm giving you 1 minute to read. If you have any doubt, please ask. Hana, everything is covered from beginning till end. You just have a look. So this is the summary you have to learn. You don't have to learn all the pages. You have to learn this. All the questions will be framed from this only. Hana, this step warriors. Please have a look. I'm giving you a few seconds. If you have any doubt, ma'am, this point is mentioned here, but you have not explained like this, something like that, you can write it down. Okay, otherwise, I think maximum is covered here. Okay, okay. Can we go ahead? So now you may be following some source now for PYQs. I mean, topic-wise question, there are many sources. You may be having subscription of some app. Maybe whatever app you are studying right now, they also provide you the question bank now. So I suggest you to follow the same question bank. And Saddam, I don't suggest you to go for topic-wise. After completing entire bacteriology, now I want you to do a mixed bag question of the entire bacteriology so that you will think in all the direction. After studying step 4 years, you are doing question of step 4 years, it is of no use. And so after studying all the 25 bacteria, if you do a mixed bag question, then you will get little bit confused and it will be more challenging. You are getting my point? Okay. Can we proceed? Does anyone have any other doubt? Or can we proceed? People, can we proceed? Huh? So have you read this? So I will not say after every bacteria, it's your duty that after every bacteria, have a look here, mark here, whatever you do, it's your choice. So you have to study these 18 pages. That's it. Not the 110 pages. Okay? Chalo. So let me come ahead. Let me come with the next bacteria, Streptococcus. Okay? I'm starting with Streptococcus. The next bacteria. So we have started GPC in the morning, gram-positive cocci. Hana, gram-positive cocci are only 2 in number. So the first test we always apply is catalase. Catalase positive, catalase negative. Catalase positive Staphylococcus, we have completed it. Hana? We have completed the 1 main hero, Staph aureus, and 2 side heroes, that is cones, Staph epidermidis and Saprophyticus. We have completed successfully. Now I'm coming on catalase negative GPC. Catalase negative GPC is Streptococcus. So let me further classify Streptococcus. So let's start the classification of Streptococcus. Please understand, everyone on your gadgets here on the screen. Please see here. So let's start Streptococcus. The first thing you do here is blood agar. Blood agar. What are the possibilities on the blood agar? I told you, in blood agar there are 3 possibilities. Some bacteria are alpha hemolytic, some are beta, some are gamma. Have I taught you this? Alpha matlab a partial hemolysis, beta matlab complete hemolysis, gamma matlab no hemolysis. I told you, है ना? What about the Staphylococcus? I told you all Staphylococcus are beta. Have I taught you? Staphylococcus के blood agar में I told you it's beta. Staph aureus is beta. All Staphylococcus are beta, I mean to say. But Streptococcus, some of them are alpha, some of them are beta, some of them are gamma. It's of 3 types. Staphylococcus, they all are beta. There is no difference in them. But Streptococcus, they can be alpha, they can be beta, they can be gamma. Hai? So alpha are only 2. Just learn their name and finish it. Only 2. Only 2. Which 2? Name them. Say, ma'am, Streptococcus pneumoniae. Pneumonia, I will teach you. Pneumonia. Streptococcus pneumoniae, that is pneumococcus, or Streptococcus viridans. Viridans. Say it out. Pneumonia and viridans. You can learn a mnemonic VP. VP. Viridans and pneumonia. These 2 are alpha. These 2 are alpha. I will teach you both. Don't worry. Gamma is only 1. Say it out and finish it. Say it out. Enterococcus. Enterococcus. So here we have E. So here we have only 2. Here we have only 1. Hana? Beta is of 20 types. 20. Yes, you heard me correct. It is of 20 types. 20 types. You will say, ma'am, what is the basis of classification? What is the difference in all these 20? You are saying it's 20 types. There may be some difference between them. So let me draw the beta hemolytic Streptococcus in front of you. Let me draw the bacteria. This is the nucleus. This is the bacteria. This is the cell wall of the bacteria. This is beta hemolytic Streptococcus. In this bacteria, in the cell wall of this bacteria, there is a carbohydrate. The name of the carbohydrate is C carbohydrate, group C carbohydrate. It's a carbohydrate present in the cell wall. It's the carbohydrate present in the cell wall. And it is of 20 types. And based on which the beta is of 20 types. So what is the basis of classification? Say how many types? You will say, ma'am, it's 20 types. 20. I will ask what is the basis of classification? You will say, ma'am, group C carbohydrate present in the cell wall, which is of 20 type, based on which it is 20 type. Okay? है ना? What is the name of these 20 type? 20 है तो 20 नाम होंगे ना? So you will say, ma'am, group A, group B, group C, group D, group E, group F. They are based on alphabet till V. है ना? A, B, C, D, E, F, G. So there is no I, there is no J. J for joker हमेशा absent होता है. I for ice cream, J for joker. These two are absent and count from A to B. You can do the counting. They are 20. है ना? Except I and J. They are 20 in number. So I will say group A, group B, group C, group D, group E, group F, group G, group H. No I, no J. Group K, group L likewise. है ना? Total 20 है. ठीक है. Okay. So 20. Got it. The basis of classification is group C carbohydrate. Got it. Their nomenclature. Got it. Who is the scientist who has done so? मिल जाए तो आज पकड़ के मारे. Who is the scientist? The name of the scientist is Lansfield. Lansfield has done this classification. Who has done this classification? Lansfield. Learn the four things. है ना? So beta is divided into 20. Say yes. The basis of classification is C carbohydrate. Say yes. The nomenclature of these 20 is A to B except IJ. Except IJ बोलने की जरूरत नहीं है. A to B, and it is done by Lansfield. Say yes. हाँ बोलो. I will not teach you 20. Obviously, I will teach you first two. Group A, Group B. In our syllabus, only two are there. So let me write the two. I will teach you Group A and I will teach you Group B. In your syllabus, that's it only. We don't have all in our syllabus. We have only two in our syllabus that we will study in detail. Okay, got it. What is the name of the Group A? Group A is known as Streptococcus pyogen, the main hero of the Streptococcus. है ना? ये यहाँ का main hero है. Streptococcus pyogen. I will teach you in much detail. And Group B is known as Streptococcus agalacti. Agalacti. है ना? I will teach you these two only. I will not teach you remaining 18. I will teach you these two only. One is pyogen, I will teach you. One is agalacti, I teach you. Say yes. The story is not over, people. है ना? Twist अभी बाकी है ना? Group A is further divided into 100 types. Group A, Streptococcus pyogenes is further divided into 100 types. This was divided in 20. This is divided in 100. Here, the basis of classification is group C carbohydrate in the cell wall. In the cell wall, there is a protein. The name of the protein is M. M for monkey, M for Mumbai, M protein. This M protein is of 100 type. So based on that, it is of 100 type. So the basis of classification is M. Please mind C is a carbohydrate, M is a protein. C and M both are present in the cell wall. C is of 20 type and M is of 100 type. Here we have done the nomenclature with A to V, the alphabets. Alphabet to khatam ho gaye. Now here we do the nomenclature with 1 to 100. So it's 1 type, 2 type, 3 type, 4 till type 100. There is no exception. ठीक है, यहाँ पे we have I and J का exception. This one was done by Lansfield. This one is done by Griffith. We are done. ठीक है, चलो, I am comparing group B. So what I taught you, start from the beginning. Start from the beginning. Streptococcus. Basis of classification is blood agar. On the blood agar, we have 3. Alpha, beta, gamma. Beta is complicated. So finish alpha and gamma first. Finish alpha and gamma first. Beta is little bit complicated. We will continue later. So first finish alpha and gamma. In alpha, we have only 2, pneumonia and viridians. Just 2. And in gamma, we have only 1, anthracoccus. There is no confusion in that. Now continue with beta. Continue with beta. Beta is of 20 types. Listen here, everyone. Beta is of 20 types. The basis of classification is group C carbohydrate in the cell wall, which is of 20 type. The nomenclature is the alphabet A to V, and it is done by Lansfield. Among them, group A is further divided into 100 types. This time, the basis of classification is M protein. This is 20. This is 100. This is group C. This is M protein. Both are in cell wall. Here, nomenclature is A to V. The alphabet. Here, the nomenclature is number 1 to 100. This is done by Lansfield. This is done by Griffith. People respond the simplest way I'm teaching you. Say, हाँ ना कुछ बोलो. Got it? Got it? So how you learn? Maximum question which you get in your exam now from group B classification. Lansfield has done classification based on C carbohydrate and Griffith has done the classification based on M protein. And in the exam, you forget that. You do ulta pulta sometimes. I'm saying Lansfield. Lansfield has done classification based on C carbohydrate into 20 types. And Griffith has done the classification based on M protein. This is a carb. This is a protein. And this is into 100 types. This you have to learn. So let's make a mnemonic. Mnemonic bana lete. ठीक है? I'm having a very good mnemonic. See if you like. LC, Lucknow City. Lucknow City. Good morning, GM. So my mnemonic is Lucknow City, good morning. Lucknow City, good morning. LCGM, है ना? Lucknow City, good morning. So Lansfield has done the classification based on C carbohydrate into 20 type. Griffith has done the classification based on M protein into 100 type. Do you have any confusion in that? I tried my best to explain you. This is how we have to learn. ठीक है? So can we say the classification once more? You want me to say the, so this is the classification written in front of you. See everyone on the screen. So we have streptococcus. We have divided the streptococcus on blood agar. We have divided in three category: alpha, beta, gamma. I know alpha, you know the two. Here it's not written, you know. Streptococcus pneumoniae, Streptococcus viridans. We know that. I know. Gamma is Enterococcus. It's written here, Enterococcus. Let's continue beta. Beta we divide into 20 types based on C carbohydrate. It is done by Griffith and it's A to B. Except IJ, it's E to B. है ना? Group A is Streptococcus pyogenes. It is further divided into 100 types. This classification is done based on M protein. This is done by Griffith and the number is 1 to 100. Do you have any confusion? Does anyone want me to repeat it or I am done? I mean, you got it. So please learn the mnemonic Lucknow City, good morning. You will never forget. है ना? Lucknow City, good morning. Hannah, learn like this. So Lansfield has done classification based on C carbohydrate into 20. Hannah, Lucknow City, 20. Good morning, 100. You can learn like this also. Griffith based on M protein into 100 types. ठीक है, alpha, beta, gamma, you know. Hannah, you can see the diagram, the cell wall. Can you see the cell wall of the bacteria? This is beta hemolytic streptococcus. Can you see the cell wall? Cell wall, can you see this purple color thing? This is group C carbohydrate. It is of 20 type. So Lansfield is classified based on this protein. And can you see the green color? The green color is the M protein. It is of 100 type, based on which Griffith has classified. Hana? People, everyone say. You got it? So basically, I'm going to teach you 5 streptococcus in this chapter, one by one. मैं पांचों पढ़ाऊंगी. In alpha, I will teach you 2. Hana? Pneumonia and viridians. PV, VP, whatever. Pneumonia, viridians, I will teach you both. Here, pneumonia is the main hero. Main hero pneumonia है. Pneumonia थोड़ा detail में देखेंगे. Viridians 2-3 line. Only 2-3 lines about that. है ना? Beta में I will teach you. There are 20. There are 20 beta. I cannot teach you 20. I will teach you first 2. Group A, Group B. Group A is pyogen, the main hero. And Group B, A galacti. 2-3 lines for that. So here main hero is pneumonia and 2-3 lines for the viridians. Here main hero is pyogen, 2-3 lines for the A galacti. Gamma is only 2-3 lines I will teach you. Not important. It's side hero only. So total 5. So I am teaching you streptococcus. In the streptococcus, I will teach you alpha, beta and gamma. In the alpha, I teach you 2, Streptococcus pneumoniae, Streptococcus viridans. Beta, there are 20. There are 20, but I will teach you only 2. Group A, Group B. The name of the group A is Streptococcus pyogenes. Here, Streptococcus, क्या कहते हैं? A galacti. ठीक है? And here it is Enterococcus. ठीक है? No, you don't have to learn the names itself, Elizabeth. You just have to learn the name of 2 only. ठीक है जी? है ना? So, in your chart, I have given the 2 main hero. Here 2 main heroes are there. So from alpha, who's the main hero? From alpha, and from beta, from alpha the main hero is this. From beta the main hero is this. Gamma is not at all main hero. So I have given the 2 main heroes in the chart. One is pyogenes. It is given in the central column, and one is pneumonia, pneumococcus. It is given here last. So Staph aureus to ho gaya, and 2 streptococcus I have given. The 3 side hero you can write 2-3 lines of them like that only. No need of the chart. If you are following the chart at all, I have told you what I'm going to do. Say yes. So what is my sequence? First, I will finish beta. In the beta, group A and group B. Then I will come on alpha. Both pneumonia and viridians. Then finally, I will come on gamma. And then I will summarize everything. Okay? So let's start with beta. Let's start with beta. In the beta, I'm starting with the first one. Group A beta, Streptococcus pyogenes. Let's start the main hero. Streptococcus pyogenes. Streptococcus pyogenes. That is group A. Group A from the beta. ठीक है? सबसे पहले हम देखते हैं culture media. On culture media, obviously it's beta. No need to say. ये beta वाले में ही तो आ रहा है. We are studying beta only. So obviously on blood agar it's beta. It's showing beta hemolysis. है ना? Selective media you have to learn too. On blood agar everyone grow na? On blood agar. So if you add crystal wallet in the blood agar, only this bacteria will grow, others will not grow. Crystal wallet blood agar. Number 1. Number 2, if you add PNF in the blood agar, what is PNF? Polymyxin, neomycin, fusidic acid. Polymyxin, neomycin and fusidic acid. So these 3 things if you add in the blood agar, again it become only Streptococcus pyogenes will grow, other will not grow. My point is that on blood agar everyone grow. Streptococcus pyogenes also grow. But add 2 things in the blood agar. Either you add crystal wallet in the blood agar or you add PNF. What is PNF? You have to learn the full form. Polymyxin, neomycin and fusidic acid. So all other bacteria are killed except Streptococcus pyogenes. If you are adding any of the two, only Streptococcus pyogenes will survive, other will not survive. Learn the two selective media. Crystal violet blood agar, PNF blood agar. Say it out. Say, ma'am, it's crystal violet blood agar. And it's PNF blood agar. Both are blood agar only. Both are blood agar. But we are adding either crystal violet or PNF. So it becomes selective. Crystal violet, PNF. Write down the two selective media, crystal violet and PNF. Can we revise the selective media of Staph aureus and Streptococcus pyogenes? I already taught you Staph aureus and currently I am teaching you Streptococcus pyogenes. Tell me the selective media, वहां पे भी 3 पढ़े थे, यहाँ पे 2 हैं. So वहां पे था Ludlam, if you remember. वहां पे था mannitol salt, है ना, if you remember. और वहां पे तीसरा था Columbia, if you remember. Please learn the composition also. Here we are having only 2. Crystal violet blood agar and PNF blood agar. Both are blood agar. You have to learn. I have given you a table of all 25 bacteria. Compare that table. Open that table and table में you can mark bacteria by bacteria. तो पूरी table भी हो जाएगी. End में you have to revise the table. Okay जी? Can we proceed? Say yes, है ना? And it's a delicate darling. Who is a delicate darling? I told you yesterday there are certain bacteria. You do home collection. This is the laboratory. So the time taken for the transit, they die. They are very delicate. One of them is Streptococcus pyogenes. So you require transport media for them. Have I taught you transport media? You require transport media for them. So your transport media is Spikes media. Yesterday I taught you for Vibrio cholerae it was Venkataramana Ramakrishnan. Have you remembered? याद आया? So here it was Spike media. मैंने लिखवाया था कल अलग से. If you remember and awaken at that time. So transport media. I don't told you transport media and Staph aureus. It was not delicate, darling, but this one is delicate. That's why we require transport media here. So we are done with the culture media. I am teaching you what? Remain oriented. पढ़ क्या रहे हैं? Streptococcus pyogen. है ना? Is it alpha? No. Is it beta? Yes. So I am teaching you beta Streptococcus. Alpha and gamma बाद में पढ़ेंगे. I am teaching you beta. Beta में it is group A. There are 20 beta. I am teaching you group A. है ना? For this I taught you the 3 culture media. I taught you what happened on blood agar. You tell me. What is the selective media? You tell me. What is the transport media? You tell me. ठीक है? Blood agar में obviously it's beta. Obviously. है ना? Selective में I told you 2 names. Crystal violet and PNF. And transport में learn it's spike. है ना? I'm taking many efforts that you learn in the class itself. ठीक है? We are done with the culture media. Now tell me what we are studying. Alpha, beta, gamma. There are 3 types of Streptococcus. Which one we are studying right now? Say, ma'am, we are studying beta right now. है ना? How many types of beta are there? Say, ma'am, there are 20 types of beta. ठीक है? So, which one we are studying? We are studying currently group A. Currently, we are studying group A. है ना? So, from group to B to V, the 19 are remaining. है ना? Out of 20, the first we are studying 19 or more. We will not study 19. We will study one more only. ठीक है? I mean to say, tell me 2 unique properties which is present only in A, not in B to B. है ना? कोई एक ऐसी property बताओ, 2 properties which is present only in group A, not in remaining 19 groups. Tell me 2 unique properties of Streptococcus pyogenes which is not present in any bacteria in this world. The 2 unique properties. So write down the 2 unique properties. ये unique properties पे ही सारे questions आते हैं. You have to learn it by heart thoroughly. Number 1 unique property, there is a special test which is known as PYR test. So this one is PYR positive. No other bacteria in this world give this test positivity. Only this bacteria. Which bacteria we are studying? Streptococcus pyogenes, group A. It gives PYR positivity, no other bacteria. And bacitracin is a chemical. Like novobacin, like penicillin, like cephalosporin, like sulfonamide. It is very डरपोक. I told you ना, it's delicate darling. इसको देखो transport media भी लगता है, बड़ा नाजुक है हिंदी में कहें तो. It's very delicate, है ना? That's why it's required transport media also. It's very delicate. You put bacitracin, it is killed. It is killed. None of the other 19. There are 20 beta, ना? 20 में से only group A, है ना? Not B to B. Only group A is killed by bacitracin. This is bacitracin sensitive. It is very delicate, darling. Nazuk kali hai na? But these all are bacitracin resistant, the remaining 19. So this is how we can differentiate A from the remaining 19. If you get a beta, then look for A, look for bacitracin sensitivity. If it is sensitive, it's A. If it is resistant, it can be anyone among the 19. Say yes, people, say yes. So I am summarizing. I am teaching you not alpha, not gamma. I am teaching you beta. Beta Streptococcus, which are of 20 types. So I have splitted the 20 into 2 groups, group A and group B to B. So here we have 1, here we have remaining only 19. I told you 2 property. Only A is PYR positive, remaining all are PYR negative. I told you only this one is bacitracin sensitive. The remaining all are bacitracin resistant. Please learn it, understand it, and learn it. Hello. So this is the thing. Learn the two unique properties. Now coming on the toxins of Streptococcus pyogenes, toxins, and followed by I will tell you the diseases it is causing. So first see the toxins. How many toxins it secretes? Only two toxins. One is hemolysin, one is pyogenic toxin. Only two toxins are there. One is hemolysin and one is pyro. Not pyo, it is pyro. The name itself depicts its meaning. मैं हमेशा कहती हूँ माइक्रोबायोलॉजी तो पढ़ने की जरूरत ही नहीं है. You just split the word, you get the meaning. ठीक है? Hemolysin. वो था cytolysin वहां पे. Here hemo मतलब blood cell. So it will split. It will cause the rupture of the blood cell. Hemolysin. So it will cause anemia. है ना? It will cause rupture of the blood cell. Hemolysin toxin. Hemolysin toxin. ठीक है? And the second is pyro. The word pyro मतलब fever. कहते हैं ना fever आ गया है. Pyrogenic. Pyro मतलब it is causing increased temperature. है ना? Pyrometer, pyrometry, increased temperature. It causes fever. So these toxins causes rupture of blood cell and these causes fever. There are two type of toxin. First say the name. First say the name of the two toxin. There in Step aureus, how many toxins we have? I'm teaching you Streptococcus pyogenes here. There we have four toxin if you remember. है ना? Cytolytic toxin, enterotoxin, toxic shock syndrome toxin, and epidermolytic toxin. बार-बार रिवाइज नहीं कहूंगी, है ना? Here we have only two toxin. Which two toxin here we have? Hemolysin, है ना? Which cause rupture of blood cell. We don't have hemolysins there. And here we have pyrogenic toxin, which cause increased temperature, which cause fever. So compare the toxin, है ना? वहां से कौन सुपर एंटीजन था? वहां से कौन सुपर एंटीजन था? These three were super antigen वहां से, और यहाँ से only this one is super antigen. Now check your column of the 10 bacteria which are super antigen, 10 organisms which are super antigen. Yesterday I have given you the list of super antigen, 5 bacteria, 4 virus, 1 fungus. Check in that. So I have mentioned Staph aureus, these three toxin. I have mentioned Streptococcus, only this toxin. So you have to learn super antigen कौन सा है. So pyrogenic is super antigen here. This toxin is not important, hemolysin. Pyrogenic is more important. All the diseases is caused by pyrogenic only. This one causes halka fulka anemia, a mild anemia, nothing else. So not very important. But pyrogenic is very important. Clinically, I mean. है ना? It's very important. Pyrogenic is very important. Please understand. Pyrogenic is super antigen first thing. It is not a super antigen. I told you. This one is super antigen. Pyrogenic causes vasodilatation of the blood vessels in the body. See. Complete body blood vessel, cutaneous blood vessel vasodilatation. Who is causing? Is it hemolysin? Say no, ma'am. It's pyrogenic. It is causing vasodilatation in the complete blood body. Hana? And the disease is known as scarlet fever. Disease is known as scarlet fever. The complete body becomes red. Imagine inside my body, Streptococcus pyogenes is entering. Hana? It is secreting a toxin. It will secrete both the toxin in my blood. But this is not very important, but this one is important. Pyrogenic will cause vasodilatation in my cutaneous blood vessel. So complete body will have a rash. The name of the disease is scarlet fever. Say, people say, maximum vasodilatation occurs in the tongue. It occurs in the tongue. Maximum vasodilatation occurs in the tongue. The tongue will become red, super red in color. It looks like the fruit strawberry. That's why known as strawberry tongue. So दोनों का reason एक ही है, scarlet fever, strawberry tongue. दोनों का reason एक ही है, vasodilatation. Here cutaneous blood vessel vasodilatation, here tongue ka blood vessel vasodilatation. So it causes scarlet fever. Here only you can write it causes strawberry tongue. Strawberry tongue, yes. It causes scarlet fever, it causes strawberry tongue. It is erythrogenic. Erythrogenic means redness. It causes redness because of vasodilatation. You got my point? Say yes. Anna. So this is the Kawasaki also have strawberry tongue. Yes, absolutely right, Sasika. But here we are not dealing with Kawasaki, but yeah, it also has strawberry tongue. You are absolutely right. And here we are having the strawberry tongue in Streptococcus pyogenes. And apart from that, it also causes toxic shock syndrome. It also causes, but here toxic shock syndrome is not very important. It is more important there in Staph aureus. Staph aureus का कौन सा toxin करा रहा था? It was toxic shock syndrome toxin. और इसका कौन सा toxin करा रहा है? It is caused by pyrogenic toxin. है ना? Now I'm interested in the two tests. One is Dick test. One is Schultz-Charlton phenomenon. What do you mean by that? Listen. Which toxin we are talking about? We are talking about pyrogenic toxin. ठीक है? Pyrogenic toxin is of three type. है ना? A, B, C. SPE, A, B, C. It's written here also. It's of three type A, B, C. What is SPE? Streptococcus pyrogenic exotoxin. Streptococcus pyrogenic exotoxin. It's pyrogenic toxin only. So A maximum causes vasodilatation. So this toxin is available commercially. What do? You take the toxin in a syringe. Which toxin? It is type A, pyrogenic toxin. It's available commercially and inject it intradermally. not intramuscular, inject it intradermally. The point where you are injecting now, that area will become red in color because of the vasodilatation. So it is proven that this vasodilatation is caused by this toxin only. And here also we are proving it in vivo, we are proving it. This test is known as dick test. How many of you got it? Dick test का मतलब, हाँ ना, dick test में क्या करना है? Take a syringe, fill the pyrogenic toxin in that. It's available commercially. Fill it, give intradermal injection, and notice the redness. It is causing vasodilatation. So naturally also in the body, it is only the way who is causing vasodilatation. We have proved that. Say people, yes, haan to bolo. Got it. This is dick test. Dick test. Schultz-Charton test is opposite of that. Hana? Here dick test we are performing in a healthy people, in a healthy individual. So on injecting toxin, redness occur. Hana? Schultz-Charton is done not in healthy people. It is done in scarlet fever patient. A patient of scarlet fever. The patient is already red. You don't inject toxin, you inject antitoxin. So जिस point पे antitoxin डालोगे, उस point पे redness चली जाएगी. The redness will disappear from that point. It is known as blanching. The redness will disappear. It is Schultz-Charton. My point is that inject toxin, the redness will come. Redness will come. है ना? Inject toxin in a healthy people. Redness will come. है ना? And inject antitoxin. Antitoxin in a person who is already have the scarlet fever. The person is red. Scarlet fever. है ना? So on doing so, the redness will disappear. Say yes, I'm trying very hard. This test is known as Dick test and this test is known as Schultz-Charton, जो भी नाम है उसका, Schultz-Charton phenomenon. Hey, Hannah, you have to identify the option. You don't have to write it. Hannah, so these are the two tests. Okay, here we are injecting toxin. Here we are injecting antitoxin. Here in a healthy people, so redness will occur. Here in a person with scarlet fever, redness disappear. Both the tests are proving that ultimately vasodilatation is caused by pyrogenic toxin. Because on injecting toxin and antitoxin of the pyrogenic toxin only, these things are happening. So we have proven it, that this toxin is the only toxin which is causing vasodilatation. Anna? So we are done with the toxins. I am teaching you Streptococcus pyogenes. Okay. I am teaching you toxins here. Okay. Anna? So I taught you two type of toxins. One is hemolysin, not important. Second is pyrogenic toxin. Pyrogenic toxin, many important things. Number one, it causes vasodilatation. Number one, usse bhi pehle, it is a super antigen. This is not super antigen. It causes vasodilatation. Since it causes vasodilatation, it is erythrogenic. Anna? Since it causes vasodilatation, it causes scarlet fever. Since it causes vasodilatation, it causes strawberry tongue. Okay, and it is responsible for two tests, Dick test and Schultz-Charton test. Anyone having any confusion, please say something. Okay, can we proceed? So toxins are done. Okay, toxins are done. Now coming on the diseases. How many diseases Streptococcus pyogenes causes? How many diseases Streptococcus pyogenes causes? You have to name the diseases. How many diseases does it causes? Okay. So it causes two type of diseases, suppurative and non-suppurative. In the suppurative, the pus is formed. In the non-suppurative, no pus is formed. Naam mein hai non-suppurative, no pus. So I will explain you. In the suppurative, we have two. In the non-suppurative, we have two. Total 4 diseases. 2 plus 2. है ना? ये others इतना important नहीं है. ठीक है? So here 2 are respiratory and skin. And here 2 are rheumatic fever and PSGN, post-streptococcal glomerulonephritis. I will explain all 4. Can I start? Can I start? Are you people there? Live, alive, awaken. Live ho, zinda ho, jag rahe ho. Do I score too much? Am I too rude sometime? No, na? So, okay. So, let's start. So, okay. So, the two types of the toxins, suppurative and non-suppurative. Okay, ji? So, suppurative is respiratory and skin. Non-suppurative, it's rheumatic fever and PSGN. One by one we will see. So, suppurative में respiratory. Respiratory, it causes sore throat. हम सबको हुआ है आज तक. We all have the sore throat till in our life. I don't think any one person don't have sore throat till date. ऐसा तो हो नहीं सकता. It's very common, very common. Sore throat होता रहता है ना. You can see pus in the tonsil. You can see patechi is there on the palate. It is known as sore throat. Sore throat. It is caused by this. The most common bacteria which causes sore throat is Streptococcus pyogenes. We all have this. Currently, I am also having it. So Streptococcus pyogenes causes sore throat. Okay. Streptococcus pyogenes is the most common bacteria which causes sore throat. Never forget. Okay. The next is the skin infection. The next is the skin infection. Skin infection, there are many, but 2 are ultra important. The 2 which are ultra important is impetigo and erysipelas. Impetigo and erysipelas. Impetigo में crust-like lesion is formed. It causes a crust-like lesion. है ना? ऐसा लग रहा है मधुमक्खी का छत्ता. It looks like honeycomb appearance in look wise. In external appearance, it is looking like honeycomb. It is looking like honeycomb, crust-like appearance. Got it? And second, it can enter in the lymphatics. It can enter in the lymphatics. It causes pus in the lymphatic. I'm saying pus, not in blood, not in skin. Pus and lymphatic. Very unique. It forms pus in the lymphatic. So that skin where the pus is formed in the lymphatic, that skin become very hard. Have you seen orange fruit? Santra. Khaya hai kabhi? Have you seen the skin of the orange? The skin, chilka. Chilka dikha hai? So it's very hard. Have you touched that? So if you close your eyes and touch the skin of this person here, you will feel like it's the skin of the orange. It's hard, it's granular. I repeat, it's hard, it's granular. So it's like and it's looking orange also. Look wise, it's also orange. It is known as PDU orange. So what are the two skin diseases, people? You will say, ma'am, the two skin diseases are impetigo, number one, and erysipelas. Erysipelas. So impetigo mein crust-like lesion is formed. And here, the pus in lymphatic is there. Here, the appearance is known as honeycombing. And here, the appearance is known as PDU orange. PDU orange. Okay, say yes. Okay, so learn the two skin. So we are done with one respiratory and two skin. Others may scarlet fever. We already know scarlet fever. So we are done with suppurative. I'm teaching you the diseases caused by Streptococcus pyogenes. I'm teaching you group A. We will come on group B also after this. Okay, I'm teaching you Streptococcus pyogenes. The first Streptococcus, I have to teach you 5 Streptococcus. ये पहला चल रहा है, 4 और पढ़ने हैं, the 4 side heroes. This is the main hero. ठीक है, 2 type of diseases it causes. What are the 2 type of diseases? Say suppurative, ma'am, and say, ma'am, non-suppurative. In the suppurative, what are the diseases? What are the options we have? Say, ma'am, there are 3 type of suppurative or 2 type. The first is the respiratory, second is skin, third is others if you want to take. ठीक है, respiratory में sore throat. Have you remembered the diagram? Skin में we have 2 diseases, impetigo, erysipelas. This is honeycombing. This is PDU orange. Others में scarlet fever. Khatam. Scarlet fever. You know the diagram of all these. I've shown you diagram of all these. You can identify an image-based question also. You can identify the sore throat, impetigo, erysipelas, scarlet fever. सबके diagram भी आते हैं. Coming on non-suppurative. Can I come on the non-suppurative? I need your permission. Can I come on the non-suppurative? It's difficult. Non-suppurative. Here, bacteria will not cause the disease. You will say, ma'am, what do you mean by bacteria do not cause the disease? Who causes the disease else? Who causes the disease else? Bacteria do not cause the disease. मतलब कौन करा रहा है disease? Let me explain. So here I will explain you 2 diseases now. Non-suppurative में one by one we will deal. First is rheumatic fever and then post-streptococcal glomerulonephritis. One by one we will see. One by one we will see that. Okay. First we will see rheumatic fever and then we will see post-streptococcal glomerulonephritis. Let me explain you rheumatic fever. Can you see this diagram? In this diagram, this is a child. Rheumatic fever occurs in child. ये एक बच्चा है, a small child, 3 to 5 years of age. This is a child. The child have tonsillitis. We also have tonsillitis whenever we were kids very frequently. So this is a tonsillitis. Tell the bacteria, most common cause of tonsillitis. Which bacteria? Which we are reading right now. That is group A. Beta streptococcus ka group A, that is Streptococcus pyogenes. So the child is having tonsillitis by this bacteria. You will say, ma'am, I also have it when I was a kid. Currently also I'm having it. So say rheumatic fever का क्या लेना देना? You are teaching us rheumatic fever ना? तो rheumatic fever कैसे हो गया? ये तो हम सबको होता है. It's very common tonsillitis. Okay, let me explain. Let me complete the story. So body is forming antibodies against the bacteria. Say yes. Body is forming antibodies against the bacteria. The antibody will go bind with the antigen, bind with the bacteria. Antigen antibody complexes will be formed. They will be phagocyzed. Bacteria is killed. You will say, ma'am, what? Bacteria is killed? Yes, the person is cured. Bacteria is killed. You will say, ma'am, okay, but where is rheumatic fever? Rheumatic fever kahan hai? Bacteria do not cause rheumatic fever. Mind my words. Take it in consideration. Mind it. Bacteria do not cause rheumatic fever. Bacteria karata hi nahi hai. ठीक है. So अब क्या आगे की story क्या? What is the story ahead? Listen, everyone. The bacteria, this bacteria, I'm teaching you Streptococcus pyogenes. It is having M protein in the cell wall. Have I told you now the basis of the Griffith classification? M protein in the cell wall. It is having the M protein. So the antibodies which are formed now, they are anti-M. They are formed against the M protein. So these antibodies which are formed, they are against the M protein of the bacteria. The problem is that human 5 organs also have M protein. 5 organ human. Can you see the 5 organs? Heart, brain, you can see the 2 vital, heart and brain. I learned like this. I have learned like this. The 2 vital, brain, heart. After that, skin. The third is the skin. You can see. Just below the skin, we have subcutaneous tissue. Subcutaneous tissue. And below that, we have joints. So say the 5 organ. Which are the 5 organ? Say, ma'am, heart, brain, skin, subcutaneous tissue, joints. फिर से बोलो, heart, brain, skin, subcutaneous tissue, and joints. These 5 organs also have M protein. See, I've drawn the M protein beautifully. See, my diagrams are very beautiful. I've done a real hard work to explain you. Listen, this is M protein on all 5 organs. So basically, the antibodies will get confused. They are formed against the M protein of the bacteria, but they think this is also bacteria. Antibody don't have eyes कि भाई ये foreign है, ये self है. उनको लगेगा this is also M protein. So they will come and react with the 5 organ, the M protein of the 5 organ, and damage the 5 organ. It's an autoimmune disease. It's an autoimmune disease. Got my point? So body's own antibody are destroying own cells. Got my point? This is known as cross reactivity. What is it known as? Actually, antibodies are not formed against the own tissue. They are formed for the bacteria. And the bacteria is a trigger. It's a stimulus. It's a trigger. After they are formed, after killing the bacteria, they're damaging the 5 organ, causing 5 diseases in 5 organs. In the heart, it causes rheumatic heart disease. In the brain, it causes chorea, Sandam's chorea. In the skin, it causes erythema marginatum. In the subcutaneous tissue, it causes nodules. And in the joints, it causes arthritis. You will study the detail in patho mein bhi padhoge, medicine mein bhi padhoge. Here I am teaching you the pathogenesis. Still, ye patho ka part hai which I am teaching you. The combination of the 5 diseases is known as rheumatic fever. My point is that rheumatic fever is not one disease. Rheumatic fever is the umbrella term given to these 5 together. Now you tell me, who is causing the 5 organ damage? Is it bacteria going to the 5 organs and damaging 5 organs? Say yes or no. Is it bacteria damaging the 5 organ? Say yes or no. Leading question. No, bacteria is not damaging. It is the antibodies. So do the pus formed in the 5 organ? No. None of the organ will have pus. No. So that's why it's a non-suppurative complication. समझ में आई बात? Why it is non-suppurative? But let me ask one more question. Can the 5 diseases, I mean the rheumatic fever, can it occur without sore throat, without bacteria? In someone's body, there is no bacteria is coming. Without that, does the 5 organs get damaged? No. If the bacteria will not come, antibodies will not form. And the 5, so bacteria ke bina bhi nahi, bacteria ke saath bhi nahi. You got my point, hai na? So neither the disease is caused by bacteria, hai na? Nor it can occur without bacteria, that I mean. Am I right? Sahi bola maine, hai na? So it cannot occur without bacteria, but it is not caused by bacteria. It is caused by the antibodies against the bacteria. Bacteria is a trigger. The disease is known as acute rheumatic fever. Say yes. It is a non-suppurative complication. Say yes. Samajh mein aayi? Got my point? The same way. The next is PSGN. One more non-suppurative complication. So I would like to draw it. See, this is a human. Let me draw a rough diagram of a human body. This is a human body. Just for explanation. This is the blood vessel. And these are the kidneys. This person also have tonsillitis. The same bacteria, which I am teaching you right now. The same bacteria. This person also have tonsillitis. By which bacteria? Name the bacteria. I am teaching you group A streptococcus. Group A beta hemolytic streptococcus. That is streptococcus pyogenes. So you will say, ma'am, okay, tonsillitis to abhi bhi hua tha. After causing tonsillitis, the bacteria is going in the blood. After going in blood, the bacteria is moving in the kidneys. Kidneys is the favorite organ. After going in the blood, kidney is the favorite organ for this bacteria. It is having affinity for the kidney. So it is moving to the kidney. In the kidney, you may be heard or you may have read it. We have a filtration barrier in the nephron. Kidney mein nephron hote hain. One kidney have one nephron. Padha hai? In the pathology. In the nephron, we have this thing. Filtration barrier. In the filtration barrier, we have a glomerular filtration membrane and we have podocytes. Have you heard about the podocytes? Podocytes hote hain. ठीक है? So bacteria go in the kidney and deposit on the podocyte. Podocyte पे जाके deposit हो जाता है. Where does it deposit? It is deposited on the podocyte. This is known as planted antigen. सुना है ये नाम पहले? Planted. The bacteria is planted there. It was not initially present. Now bacteria is cleared from the blood. There is no bacteria in the blood. It is planted on the podocyte. Now the body will make antibody for the bacteria. है ना? Antibody will go पीछे behind the bacteria. The antibody will go behind the bacteria. Jahan bacteria, wahan antibody. है ना? So wherever the bacteria is going, antibody is going there. I mean to say, antibody will also go to the podocyte and deposit there. So antigen-antibody complexes are formed and they destroy the podocyte. They destroy the filtration barrier, and the person will have glomerulonephritis. Such glomerulonephritis is known as post-streptococcal glomerulonephritis, PSGN, because it occurs after the streptococcus. Now you tell me who's destroying the kidneys. I mean, who's destroying the filtration barrier? Who's destroying the podocyte? Is it only antigen? Say no, it is not bacteria. It is the antigen-antibody complex. So here also it is non-suppurative complication. How many of you got it? कितनों को समझ में आई? Say, I'm teaching you streptococcus pyogen. है ना? So two type of diseases it causes: suppurative and non-suppurative. In the suppurative, there are two diseases: respiratory and skin. In the non-suppurative also we have two, rheumatic fever and PSGN. ठीक है? In the respiratory, the main disease is sore throat. We all know. In the skin, two diseases are there: impetigo, erysipelas, honeycombing, PD orange. है ना? Rheumatic fever and PSGN, these are not caused by bacteria. These are caused by antibodies against the bacteria. That's why these are non-suppurative. I tried my best to explain you. Appreciate it. Got it? We are done with the diseases caused by Streptococcus pyogenes. The pathogenesis is in front of you. We are done. We are done. Can we revise the pathogenesis of Strep aureus? It causes 4 diseases caused by 4 toxin. It causes pus everywhere because of cytolytic toxin. It causes food poisoning because of enterotoxin. It causes toxic shock syndrome because of toxic shock syndrome toxin. And it causes SSSS because of epidermolytic toxin. So these were the diseases. There are 4 diseases. यहाँ पे भी 4 ही हैं. There also we have 4. Here also we have 4. But you can see here 2 are suppurative, 2 are non-suppurative. Here the 4 with 4 different toxins. Okay, I tried to explain you, to understand you, to learn you. We are done with the main hero, one of them. I'm teaching you beta. I'm not teaching you alpha and gamma. Currently, I'm teaching you beta. In beta also, I have to teach you 2. Group A, Group B. Streptococcus pyogenes and Streptococcus agalacti. By the way, I'm done with this. I'm done with Group A. So we will solve the questions of Group A. This was the main hero from here. सबसे बड़ा यही था. I want you to read the chart completely. हमने complete कर लिया ये bacteria. We will solve some questions and we will move on Group B. Are you with me? Group B is very small. You have to learn only 3 or 4 lines of that. There is no chart. After that, we will move on alpha and then gamma. You got the sequence? Orientate. Read the question. Tell me the answer. You have to be fast enough to respond. सोचते नहीं बैठना है. Reflex अपना जरा fast करो. Read the question. Tell me the answer. C carbohydrate in Streptococcus hemolyticus is important for which classification? Is it Landsfield classification? Is it phagocytic inhibition? Is it toxin production? Or is it hemolysis? What's the correct answer, people? Yes, the correct answer is A. I want each and every one of you to answer. Yes, the correct answer is A. You all are right. Landsfield. I told you Lucknow city. So Landsfield classification is based on group C carbohydrate. This is group C carbohydrate. This is Landsfield classification. ठीक है, Lucknow city. Read the next question. There is a child presented with skin lesion. Now it's difficult. Please, we will take the clues. एक एक करके. You know nothing, है ना? And start from the beginning. There is a child. Clue ले लो. एक बच्चा है. मुझे अभी तक क्या समझ में आया? There is a child, है ना? Who is having some skin lesion. Many bacteria causes skin lesion, है ना? Many bacteria can cause. Culture show gram-positive cocci. Who is a gram-positive cocci? You tell me. Who is a gram-positive cocci? There are 2 gram-positive cocci. It can be staph. It can be strep. I don't know till now. I don't know till now. But the next line, gram-positive cocci in chain. Chain. Reading the word chain, I will remove staph from the consideration and I will concentrate only on strep. Because strep is in clusters and strep is in chains. But strep is of 3 type, alpha, beta, gamma. I don't know which one. Is it alpha? Is it beta? Is it gamma? मुझे नहीं पता. The next line is given, it's beta. It's beta. It's not alpha. It's not gamma. It's beta. In beta, there are 20. But in our syllabus, only 2 are there. So question, answer उन्हीं में से होगा. The 2. Which 2? Group A, Group B. Group A is Streptococcus pyogenes. Group B is Streptococcus agalacti. By the way, I have not taught you Group B till now. But I taught you something unique for A which is not present in other 19. I taught you. So what is that property? Is it bile solubility? Is it optochin sensitivity? Is it bacitracin sensitivity? Is it catalase positivity? So it's bacitracin sensitivity. You all are right. So correct answer here will be C based on this. How many of you got it? It's a clinical big question, approach based question. It's bacitracin sensitivity. Right? Read the next question. Tell me the answer. Try. Bacitracin sensitivity differentiate what from what? I told you. Bacitracin sensitivity differentiate what from what? Does it differentiate group A Streptococcus from Staphylococcus? Does it differentiate group A Streptococcus from beta hemolytic? Other beta hemolytic? Does it differentiate group A Streptococcus from other gamma? Does it differentiate group A Streptococcus from other alpha? So we have 2 GPC. Please see the approach. We have 2 GPC, staph and strep. So the test which differentiate the 2 is catalase. Catalase differentiate the staph and strep. Okay? Staph में positive, strep में negative. After that we have strep. After continue the strep. Strep में we have 3, alpha, beta, gamma. The beta is of 20 type. From which group A is different and B to B is different. है ना. So we apply bacitracin test. This one is delicate, darling, so bacitracin sensitive. Rest all are bacitracin resistant. So basically bacitracin is differentiating between what and what you tell? Group A from other beta, not other alpha, not other gamma, not staphylococcus. Got my point? So answer is B. How many of you are convinced answer is B? Not A, Sanjay, answer is B. Got it? Answer. Why answer is B? It is differentiated in group A streptococcus from other beta. The remaining beta. The remaining beta are B2V. The remaining beta are B2V. Total 20 are there. So bacitracin is separating the total 20 beta together. है ना? So we divide them into A separate and B2V separate. है ना? So this is the bacitracin. So this one is bacitracin sensitive. Remaining all are bacitracin resistant. How many of you got it? How many? So correct answer is B. I'm changing the question. If you got it, I'm changing the question. Instead of bacitracin sensitivity, I'm saying catalase test. Now you give the answer. Catalase test is used to differentiate what from what. What from what? You know catalase किसके लिए use करते हैं? Read the options very carefully and tell me the answer. Yes, very good, Canal. Very good, Saeed. Very good. Yes, Nasser, Prabingal, everyone is correct. Yes, Shibam. So correct answer will be A in that case. It separates strep and step. Any strep from any step. The point is step and strep. Catalase. So which test is differentiating what? It should be crystal clear in your mind. So correct answer is A. So out of the 5 streptococcus which I have to teach you, 2 alpha, 2 beta, 1 gamma, 1 is done. The main hero सबसे बड़ा हो गया, बाकी सब छोटे छोटे हैं. These all are small. We will complete 5 minutes on each of them. In the next 10-15 minutes we will finish the chapter. Okay जी, say yes. People say yes. Respond. Say yes. Huh? So Streptococcus A galacti. I'm starting with group B. Group B. Yes. Group A is done. Group B. How many type of beta are there? 20. But how many we have to study? Only 2. Which 2? Group A, B. A is done. And now I'm starting group B. ग्रुप बी का नाम क्या है? फर्स्ट लर्न द नेम. पहले नाम तो याद करो, दोस्ती तो कर लो, उससे फ्रेंडशिप कर लो. सो व्हाट इज द नेम ऑफ द ग्रुप बी? वन ऑफ द बीटा. One of the beta I'm going to take out of the 20. It's second, group B. The name is Streptococcus A galacti. A galacti. A galacti. है ना? The Streptococcus A galacti. ठीक है? Got it? Okay. Harsh, if it is written, please correct it. Whatever I'm teaching you, it's correct. If there is some misprint in the note, you can correct it. है ना? If Streptococcus pyogenes is given group B, you make it group A. Okay? Maybe some typing mistake. Anyways. So group B is Streptococcus A galacti. समझ में आई क्या? Group B is Streptococcus galacti. है ना? Now it causes only one disease. Learn one disease. Neonatal sepsis and meningitis. Who's a neonate? The first 28 days. है ना? First 28 days. First 28 days का बच्चा, the neonate, the newborn, up to first 28 days is a neonate. So it causes meningitis and sepsis only in neonates, not in infant, not in adult, not in children, not in old age, no other age. Neonatal. So you will typically get a question neonate. Neonate आते से this should be a differential. Please, है ना? You have to pick up the words. Neonate आते से this should be a differential. ठीक है? It causes neonatal sepsis and meningitis. And I am teaching you beta. Again, let me come alpha, beta, gamma. There are 20 type of beta. है ना? So in the 20 type, let me separate B from all others. A and C to B. The remaining 19. The remaining 19 I'm taking on one side and B is I'm taking on one side. I'm concerned with B now. So tell me 2 unique property which are present only in B, not in remaining 19. Not in even in A and not even in any other. So tell me 2 unique property. 2 unique property. One is it is hippurate positive. I will explain what is hippurate. No other bacteria is hippurate positive. Number 1. Number 2, it is CAM positive. CAM positive. CAM positive, other bacteria are there, but among these 19, no other is CAM positive. I know. So hippurate positive and CAM positive. Hippurate and CAM is something unique. है ना? Can you read these 2 terms? Bacitracin and PYR. ये दोनों उल्टा हैं. Bacitracin and PYR were the unique thing in group A. This is bacitracin resistant, not sensitive. This is PYR negative, not positive. Are you getting my point? देखो मैं फिर से बताती हूँ. Everyone who is having confusion. I am teaching you Streptococcus. Start from the beginning. There are 3 types of Streptococcus: alpha hemolytic, beta hemolytic, gamma hemolytic. The beta are 20. Everyone know beta are 20. But I want to teach you only 2. Group A and Group B. है ना? Group A and Group B. Now I taught you 2 unique properties of A which are negative in remaining 19. And I am teaching you 2 unique property of B which are negative in remaining 19. है ना? So you have to learn the 2 unique properties of each of them. Group A में unique were PYR positive and bacitracin sensitive. है ना? So these 2 are reverse here. है ना? It is neither PYR positive and bacitracin resistant is there. Sensitive नहीं है, resistant है. So यहाँ पे 2 दूसरी unique property है ना? It is hippurate positive and CAM positive. है ना? So this one is hippurate negative, camp negative, obviously. Got my point? So learn the two unique properties here to here. That's it. Learn the two unique properties. जिस पे question बनेगा. Say yes, people say yes. So what are the two unique properties? Say group B की. Say ma'am hippurate positive and camp positive. I will explain both. पहले learn तो कर लो. हाँ, first learn hippurate positive, camp positive. ठीक है? तो camp क्या होता है? One by one we will see. अच्छा चलो hippurate. Hippurate is an enzyme, hippurase. है ना? Streptococcus agalacti have an enzyme known as hippurase inside it, like catalase, like coagulase. Hippurase is an enzyme which is present only in this group B. No other bacteria have this enzyme. So hippurase will degrade the hippurate. Hippurate एक yellow chemical होता है. On degradation, it will convert into purple. So yellow to purple color change होगा hippurase से. Don't learn the test. Nobody is going to ask you भाई क्या है कैसे है. Nobody. Just learn hippurate positive. But someone of you may be thinking, na ma'am, hippurate hota kya hai? Samjhao. So this is the explanation. But you don't have to learn it. You don't have to learn it. Learn the summary. It's hippurate positive. Which one? Group B. Group B is hippurate positive. This is hippurate test. Hippurate test positivity is due to the enzyme hippurase. Hippurase. Got it? है ना? So number one. And number two, what is camp? The full form of the camp is the four scientists who created it. CAMP. C for Christie, A for Atkin, M for Munch, and P for Peverson. है ना? Don't learn the full form. Just for your knowledge, I'm telling you. Camp में four scientists हैं. Camp reaction is the four scientists. Don't learn the full form. Nobody is going to ask you that. ठीक है? Now first you tell me what I'm teaching you right now. You will say, ma'am, you are teaching us a streptococcus. The name of the streptococcus is Streptococcus agalacti. Am I teaching you that? You tell me which hemolysis does it show? Alpha, beta, gamma. कौन सा वाला पढ़ा रही हूँ मैं? Alpha वाला कि beta वाला कि gamma वाला? आप बताओ पहले. Which one I'm teaching you? I'm teaching you beta. There are 20 beta. I'm teaching you group B वाला, second beta. है ना? It show beta hemolysis. Have I taught you chapter number 1, Staph aureus? Have I taught you Staph aureus? Of course, I completed Staph aureus. है ना? You tell me Staph aureus कौन सा hemolysis दिखाता है? Alpha की beta की gamma? You tell me first. So all Staph aureus show beta. All Staph aureus show beta. Streptococcus can show alpha also, beta also, gamma also, but currently this one also show beta. So my point is that both of these bacteria show beta. है ना? Both of these bacteria show beta. I guess everyone understand. Now take a blood agar. Take a blood agar. Let's start camp. I'm explaining you camp. Understand? Now it's difficult. I will explain only once. ठीक है? Take a blood agar. ले लो blood agar ले लो. Take, है ना? Take it. Take a streak of Staph aureus. Yes, chapter 1, Staph aureus ले लो. Staph aureus. ठीक है? Perpendicular to this, take another streak of Streptococcus agalacti, जो मैं अभी पढ़ा रही हूँ. Streptococcus agalacti. Galacti. है ना? Take the perpendicular streak. You will say, ma'am, both show beta. It is blood agar, no? Blood agar पे either alpha or beta or gamma. So both of them show beta. Just now you told us. Staph always show beta, and Streptococcus में alpha, beta, gamma तीनों हो सकते हैं, but this is beta. So both start. So hemolysis start from the center. Listen my point. Hemolysis start from the center. If both of them show equal hemolysis, it should be a circle, the zone of circle. But it's not a circle. Circle नहीं बनता यहाँ पे. Listen, मेरी बात सुनो. We get more hemolysis on this axis as compared to this axis. We get more hemolysis on this axis as compared to this axis. This is known as camp positive. We get more hemolysis on agalacti axis as compared to Staph aureus axis, and because of which we get a butterfly shape, not a circle. This is butterfly. It is known as butterfly shape या arrowhead. Arrowhead, एक ही बात है. Arrowhead या butterfly. Butterfly, one and the same thing. This is camp positive. Say yes. You can see. Can you see here? This is the blood agar. Say yes. The perpendicular streak, Staph aureus. This is Staph aureus. This one, Streptococcus agalacti. ये सारी Streptococcus agalacti की है ना? Appreciate the butterflies. I want all of you to appreciate there is more hemolysis on this axis as compared to this axis. Nobody will explain you in this world this camp reaction. सबको याद करा देगा camp positive है. बस इतना याद कर लो. क्या होती है camp reaction? मैं समझा रही हूँ, है ना? I'm explaining you. है ना? I'm taking many efforts. You don't have to learn भाई क्या होता है. Nobody is going to ask in there. For your understanding, I'm teaching you. So learn the summary. It's camp positive. It's hippurate positive. Both of them are shown here. Hippurate positive, camp positive. कहानी खत्म. Which bacteria? Streptococcus agalacti. Summary is that. Learn the summary. But if I told you directly, you may be thinking, ma'am, what is all these terms? I don't understand. But now you can have a better understanding. You have seen it now. What is hippurate? What is camp? You can understand it now. So this is camp positive, hippurate positive. Learn Streptococcus agalacti. The summary is that. Okay, G. Can we go? Hana, the two unique properties. And it causes only one disease, neonatal sepsis and meningitis. We are done with the side hero also. So learn, we are done with beta hemolytic. Beta hemolytic, there are 20, but we don't study 20. We study 2. Group A, Group B. ग्रुप ए का नाम याद करो, पायोजन। ग्रुप बी का नाम याद करो, ए गैलेक्टी। ठीक है, ग्रुप ए की 2 यूनिक प्रॉपर्टीज, PyR positive, bacitracin sensitive, the 2 unique property of group A. Learn 2 unique property of group B, cam positive, hippurate positive. Learn 2 unique here, 2 unique here, है ना? If you want me to summarize the disease, यहाँ पे तो एक ही disease है, neonatal sepsis and meningitis. You will get a question, there is a neonate having Caesar मतलब meningitis. You will get a question like that, है ना? Or sepsis, high grade fever. ठीक है, here we have suppurative and non-suppurative. You know the story. Suppurative, there are 2 suppurative. Non-suppurative, there are 2 non-suppurative. You know the complete story. Got it? We are done with beta. We will solve some question on B, group B. है ना? Read the question. Take the clues slowly. Don't rush, don't jump. Everyone knows the answer. I'm not interested in answer. I'm interested in approach. तो approach समझो. Answer सबको पता है, ये गैलेक्टी ही आएगा क्योंकि वही पढ़ाया अभी, है ना? So please understand. Approach is important. Listen. There is a child. Take the clues. Age is always important. Always important. So till now I'm imagining. Whatever I read, I imagine. So there is a child having sepsis. So I imagine a child is having sepsis, having high-grade fever. The bacteria isolated show beta hemolysis on blood agar. There is some bacteria which show beta hemolysis. है ना? So, okay, beta hemolysis on blood agar, that is resistant to bacitracin. Resistant to bacitracin पढ़ के, can I remove this? Can I remove this? It is sensitive to bacitracin. Pyogen is the only bacteria which is sensitive to bacitracin. Rest all 19 are resistant. So I will remove this option. It is beta hemolytic. Beta. Streptococcus pneumoniae is alpha. Can I remove this option? Enterococcus is gamma. Can I remove this option? Huh? So see how I'm removing. What is the reason why I'm removing? The question mentioned beta hemolytic. We know enterococcus are gamma. We know pneumoniae and viridians are alpha. So I'm removing these two. Why I'm removing pyogen? Because it is sensitive to bacitracin. Here it is resistant. So till now I have removed 3 options. ठीक है, and it is cam positive. Cam positive is A galaxy. So I'm left with only one, and it is also cam positive. इससे भी double prove हो गया. So yes, my answer is B. है ना? I was more happy अगर child की जगह neonate देते तो, है ना? अगर child की जगह neonate होता क्वेश्चन में, I was very happy. Yes, 100% correct. But ठीक है, माफ कर दो, गलती हो गई बेचारों से, है ना? So they are mentioning child. It's not very good question. Ideally, it should be neonate. It's okay. ठीक है, कुछ चीजें चलानी पड़ती हैं. ठीक है, but rest everything is matching ना, except the term child, everything else is matching. So I will go with A galaxy. है ना? इस बात पे अड़ मत जाना, मैम you told us it's occurs in neonate, it's a child, तो ये आंसर नहीं होगा. It's foolish. ठीक है, थोड़ी बहुत चलता है आगे पीछे, but yeah. So this is the thing. So we are done with this question. Okay. So let's start with alpha. We are done with beta. Both the beta are done. Group A is done, Group B is done. Group A, Group B both are done. Hana? Let's start with alpha with full energy. Let's start with alpha. I'm going to teach you 2 alpha. Pneumoniae and viridians. One by one. I will teach you 2 alpha. Okay? Okay, ji? Chalo? So let's start with alpha. Can you see my screen? Someone is saying the screen is black. I will give you a fast break. Let me finish the streptococcus. I'm in the middle of something. How can I give you the break? Let me finish the streptococcus. Huh? Another 10-15 minutes. Let me finish it, then we will take the break. Oh, मुझे break की नहीं पड़ी, तुम सबको बड़ी पड़ी है. You just eat now while watching. I will give. Wait a while. Let me finish the streptococcus, then I will give. Okay? Because if I give the break right now, the link will be, broken down. So 2 streptococcus are done, 3 are more. They are small. Let's finish them. So let's start with alpha. G. Let's start with alpha. I have to teach you 2 alpha, pneumonia and viridians. Pneumonia थोड़ा सा बड़ा है, not very much. Let's start with pneumonia. Let's start with streptococcus pneumoniae. I'm teaching you streptococcus, alpha, beta, gamma. The 2 beta are done. I'm coming on alpha. In alpha, I have to teach you 2 pneumonia and viridians. I'm starting with pneumonia. ठीक है, pneumonia की खास बात, pneumonia. पहली बात, it's a cocci, है ना? But actually, it's not a circle. It's flame shaped. All the cocci are like circles. It's not a circle. No, it's not a circle. It's flame shaped. Pneumonia is flame shaped. Number 1. Number 2, it is in pair. Number two, it's always in pair. Pair, जोड़ी में होता है. इसको अकेले रहना पसंद नहीं. It's always in pair. So number 1, it's flame shaped. It's visible. Number 2, it's in pair. Because it is in pair, it is known as diplococcus. Diplococcus means it's always in pair. Number 3, one pair is surrounded by one capsule. It is a capsulated bacteria. But capsule is not around each bacteria. Mind my words. One pair have one capsule. Tell me the mnemonic of capsulated organism. The 9 capsulated organism. Yes, very killer bacteria have pretty nice capsule. You know the 9. ये 9 है ना? P for pneumococcus. So I will prove. मैंने बोला था ना कल ये 9 बार आएगा. जब आएगा तब बताऊंगी. So it is proven that it's a capsulated bacteria. So this is the morphology. Learn three things. It is flame shaped, it is in pair, it is diplococcus, one capsule around each pair. Learn it. है ना? Based on the capsule, it is of 95 type. Based on the capsule. You can see the morphology in the diagram also here. You can see they are flames. You can see they are in pairs. You can see one capsule around each pair. One capsule around each pair. It is diplococcus. Diplococcus. Flame shaped or lance shaped. Lance भी बोल सकते हो. Lanceolate shape. है ना? You can see. One capsule around each pair. Based on the capsule, it is of 95 types. This is the morphology. है ना? Capsule demonstration. Yesterday also I taught you two ways. Who was present in my class yesterday? Huh? Have I taught you two ways for capsule? Number one, negative strain, India ink. Strain the background. Stay in the background, not the bacteria. So a capsulated bacteria will stand out in a negative background. Say yes. Huh? So negative stain. Number one. Number two, have I taught you Quill-Lung reaction yesterday? Have I taught you what is Quill-Lung? I'm explaining again. Listen, this is the bacteria, flame shaped. Huh? This is the bacteria, flame shaped. Okay, flame shaped. Here is the capsule, but capsule is very small. I can't see it. I want to see the capsule. So I will swell the capsule. I want to swell the capsule. What I want to do? I want to swell the capsule. The point is that how should I swell the capsule? If the capsule is swollen, I will be able to see that. How to swell the capsule? React it with antibodies against the capsule. It is known as anti-sera. Anti-sera. Anti-sera is antibodies against the capsule. So antigen-antibody reaction will take place and the capsule will swell. Swelling of the capsule is known as Quill-Lung. Quill-Lung reaction. And you can see it now. So this is Quill-Lung. See the capsule here? Appreciate it? Just a second. Appreciate the capsule here. Appreciate the capsule here. Can you appreciate the swelling? The swelling is due to antisera. Say yes, Quill-Lung. So learn the two ways of capsule demonstration. Negative strain, Quill-Lung. Negative strain, Quill-Lung. You get questions on that. Okay. Now here selective media is only one blood agar. There is no alag se selective media. I told you Staph aureus ke 3 selective media, Ludlam, mannitol salt, Columbia. I told you Streptococcus pyogenes की 2 selective media, crystal violet and PNF. याद है? Now I'm teaching you pneumococcus, Streptococcus pneumoniae, pneumococcus. Here we have simple blood agar. We don't have special agar. We have blood agar. कौन सा blood agar? Sheep का blood agar. है ना? Take sheep blood agar. You tell me on blood agar, will it show alpha or beta or gamma? Will it show what? Alpha or beta or gamma? I'm teaching you Streptococcus pneumoniae. Pneumonia कहाँ आता है? Alpha में कि beta में कि gamma है? There are 3 types of Streptococcus ना? कहाँ पे आता है? तो of course it is alpha. I'm teaching you right now alpha. So it will show alpha hemolysis. Everyone knows it's alpha. Everyone. No need to learn it's alpha. Alpha important नहीं है. Alpha hemolysis तो मिलेगा. So these are the colonies. Colonies के around greenish zone is not visible. लेकिन थोड़ी देर बाद greenish zone आने लगेगा. Around the colony green zone. Alpha. ठीक है, everyone understand. After some time, there is a hole at the center of each colony. There is a hole. There is a depression. There is a hole or depression at the center of each colony. Can you see? Have you ever played carom? Carom खेला है कभी? I still play every day with my kid. है ना? So carom. Have you seen the carom coin? Carom की गोटी देखी है? So carom coin के center में there is a depression. There is a depression at every carom coin. So the colony appearance is looking like the carom coin. It is known as carom coin appearance. What is known as carom coin appearance? What it is known as? Carom coin appearance, also known as Draughtsman appearance. What is known as Draughtsman? One and the same thing. So please learn Streptococcus pneumoniae on blood agar give carom coin or Draughtsman appearance. What does it give? Carom coin or Draughtsman appearance. Please learn. So at the center there is a depression. At the center there is a depression. Got my point? Anna? Now 3 unique properties which are present only here. Alpha, beta, gamma. Anna? There are 2 alpha. There are 2 alpha. Streptococcus pneumoniae and Streptococcus viridans. There are 20 beta and there is 1 gamma. So tell me something unique here which is not present in any other bacteria of the world. Tell me something very unique. You have to learn the unique properties of all the bacteria. Based on which your all questions will be there. So pneumonia ki unique property. If you ask that unique property of pneumonia, I remember where is your bank account, in which bank? Pneumonia have bank account in Bank of India. The mnemonic is BOI, Bank of India. Please, pneumonia से मुझे Bank of India याद आता है. If you say ma'am pneumonia, I will say Bank of India. So please learn. Pneumonia का bank account कहाँ है? Bank of India. What is the full form of Bank of India? The 3 unique properties. BOI, BOI, Bank of India. You can learn any mnemonic. ठीक है? B is bile solubility. Only this one is bile soluble. No other bacteria in this world is bile soluble. I will explain what does it mean. O is optogen sensitivity. Only this is optogen sensitive. Rest all, none of the bacteria is optogen sensitive. And I is inulin fermentation. I will explain. Can you see the blood agar? Say yes. On the blood agar, can you see the colonies? These are the colonies. These all are the colonies on the blood agar. These all are the colonies on the blood agar. Where are the colonies? यहाँ की colonies कहाँ गई? They disappear. You put few drop of the bile. यहाँ पे bile का एक drop डाल दो. The colony dissolve. This is known as bile solubility. The colonies get dissolved in the bile. The colony get dissolved in the bile. It get disappear when you put bile. Only this bacteria show this property. No other bacteria in this world show this property. You may be thinking the reason for that, ma'am. Where is the colony gone? Why does it disappear on putting bile? Because this is the diagram of Streptococcus pneumoniae. है ना? In the cytoplasm of this bacteria, there is an enzyme. The name of the enzyme is amylase. What is the name of that enzyme? Amylase. Amylase is suicide enzyme. It is suicide enzyme. It causes rupture of the bacteria. It is a suicide enzyme. It causes rupture of the bacteria. So normally it is inactive. But once you put the bile, na, in presence of bile, the amylase get activated and it will cause the rupture. You got my point? It will cause a rupture. Say yes. Got it? So that is bile solubility. That is the meaning of the bile solubility. Okay. So bile solubility is due to this central depression. You didn't ask the reason. You should ask the reason. Ma'am, why there is a central depression after some time? Central depression का भी reason यही है. Amylase get activated. After some time, the amylase get activated, the suicide enzyme, and central bacteria का सबसे पहले activate होगा. That's why central bacteria get disappear, and gradually it will come to the periphery. So reason for central depression and reason for bile solubility, दोनों का reason है amylase, the suicide enzyme. Say people, yes, say, है ना? O is optocin. Optocin is a chemical. Novobacin भी एक chemical था, है ना? Bacitracin भी एक chemical था, and now optocin. Optocin से सिर्फ एक बैक्टीरिया डरता है दुनिया में. Only one bacteria get afraid of optocin and get killed by the optocin. That is pneumococci. No other bacteria in this world is killed by optocin. This one is optocin sensitive. No other bacteria is killed by optocin. And last is inulin. Inulin is a sugar. It's not insulin. It's inulin. It's like glucose, fructose, galactose. ये एक sugar है ना? So it's a unique sugar which is fermented only by pneumococci. No other bacteria ferment inulin. So tell me the three unique property of pneumococci. Tell me the three unique properties of pneumococci. Pneumococci से मुझे याद आता है Bank of India. The three unique properties. Say the full form, people. Bile solubility. Imagine bile solubility is due to amylase. Amylase का activation. The colony disappear on putting bile. Optocin sensitivity. Optocin is a chemical that kills this bacteria. And inulin fermentation. Inulin is a sugar which is fermented by. So only pneumococcus show the three properties. None of the other bacteria in this world show these three properties. Everyone got it? So that is pneumococcus. Disease नाम में ही है. क्या पढ़ा रही हूँ मैं? नाम बोलो पहले. Pneumococcus, Streptococcus pneumoniae, pneumococcus, या Streptococcus pneumoniae. So obviously it will cause pneumonia. No need to say. Pneumococcus cause pneumonia. Both pneumonia, lobar and lobular. It cause both type of pneumonia. तो pneumonia कराता है. Nobody is going to ask you that. It's very easy. Pneumonia causes meningitis also, that you have to learn. So it causes two diseases. Pneumonia causes pneumonia. Everybody knows that. Even a KG student will say कि हाँ pneumonia causes pneumonia. Nobody is going to ask you that. है ना? Pneumonia causes meningitis also, that you have to learn. So it causes two diseases, pneumonia and meningitis. Learn the two diseases. We are done. है ना? So I taught you pneumonia, Streptococcus pneumoniae. So alpha, beta, gamma. We are done with the two beta. In the alpha, I am teaching you one of the alpha, Streptococcus pneumoniae. है ना? I taught you the culture media. Say yes. है ना? You have to tell me the culture media. Special एक ही culture media है, blood agar. Blood agar पे something unique. है ना? You have to tell me the morphology उससे भी पहले. है ना? There is something unique in the morphology. है ना? You have to tell me the culture media. है ना? And you have to tell me the diseases caused by that. No. You have to tell me the unique properties also. The biochemical properties. Unique properties and then the diseases. That's it. इतना ही तो पढ़ाया है. Morphology में you will say, ma'am, it's flame shaped, it's in pair, it's diplococcus and it's capsulated. One capsule around each pair. Based on capsule, it's 95-10. As simple as that. ठीक है? Culture media में blood agar में you will say, ma'am, carom coin appearance या फिर Drodsman appearance. है ना? तो central depression in the colony. The reason for the depression is amylase. सारे question cover करते जाओ. है ना? The three unique properties you all know. Bank of India, BOE, bile solubility, optocin sensitivity, inulin fermentation. ठीक है? Diseases, it causes only two diseases. Pneumonia causes pneumonia. Everyone knows that. But pneumonia causes meningitis also, that you have to learn. Okay? Picture will be same, Sasika. Sasika is asking bacitracin and optocin. Picture is same. So you want me to explain this picture? This picture is not for the chemical, Sasika. You didn't got it. This is antibiotic susceptibility test. Listen, you take a blood agar. You take a filter paper. Okay, let me take two blood agars. This is a blood agar, this is a blood agar. You take two filter paper, circular cut filter paper. This is a filter paper dipped in optogen. This is also a filter paper dipped in optogen. So I have put a filter paper at the center of the blood agar, which is dipped in optogen. So optogen. Not try to grow the bacteria. Not try to grow whatever bacteria, the lawn culture of the bacteria. Now either the bacteria will touch the rim of the filter paper and grow everywhere. Touching the rim, grow everywhere. Let me fill it. Filler. Let me fill. These are the bacterial colonies everywhere. It's a lawn culture. है ना lawn culture पता है ना हर जगह bacterial growth is there. The point is that it is touching the rim. I mean to say it is touching the rim. So it is not afraid of optogen. It is not killed by the optogen. The second possibility, the bacteria is not coming closer to the optogen. It is forming the colonies at the periphery, but not coming closer to the optogen. Got my point? So I will say, ma'am, this bacteria is optogen. This bacteria is optogen resistant. ये ऑप्टोजन रेजिस्टेंट है. इट इज नॉट अफ्रेड. ये तो ऑप्टोजन को आके टच कर रहा है. इट इज टचिंग द ऑप्टोजन and this bacteria is not coming closer to the optogen. Because it will come closer, it will die. So this bacteria is optogen sensitive. Got my point? है ना? So this is how we differentiate. This zone is known as zone of inhibition. In the zone of inhibition, you don't get any colony. Got it? So not only optogen. If you want to check the bacitracin, take the filter paper in the bacitracin. है ना? This will be bacitracin. If you want to test novobacin, take the filter paper in novobacin. So diagram तो हमेशा यही रहेगा ससिका. This one. This one is a diagram of sensitivity. This one is a diagram of resistance. है ना? The chemical changes, the filter paper changes. I hope I'm able to explain you. Got it? How many of you got it? So can you see this diagram? ये मैंने diagram दिया है optogen sensitive. ये diagram. Can you see the filter paper at the center? Appreciate the filter paper. This filter paper is dipped in optogen. So I don't know, image is very not clear to you, but in this zone there is no colony. There is no colony, and the colonies are present apart from. So this one is optogen sensitive. I mean to say, optogen को replace है bacitracin, तो diagram तो यही रहेगा. Say yes, I can replace with novobacin also. Got it, everyone? This is sensitivity test we do so. है ना? So chalo, read the question, tell me the answer. We are done with this bacteria also. Read the question, tell me the answer. There is a 9-year-old child presented to the OPD with a complaint of fever, vomiting and seizure. Chalo, take the clue. The child is 9 and having seizure. Seizure in a child, they are talking about meningitis. जब भी क्वेश्चन में सीजर आए, they are talking about meningitis. Seizure in a child, meningitis. So they are talking something about meningitis. The gram stain is given to you. Ruko, wait. Whenever gram stain is given to you, you have to see two things: shape and color. बिना shape color के proceed मत करना. Tell me the color, tell me the shape. You people are not color blind, right? Tell me the color. Is it violet or red? यहाँ पे जो बैक्टीरिया दिख रहे हैं, these small bacteria, they are violet or red? I'm asking. Are they violet or they are red? Of course they are violet. You can see they all are violet. Since they are violet, it's gram positive. It's gram positive. And are they cocci or bacilli? You can see the circle or you can see the elongated bands. I can see small circles are there. Small circles are there. So it's a gram positive cocci. So this is the approach. I will jump approach by approach. So I can see violet, that's why gram positive. I can see cocci, circles, that's why cocci. Gram positive cocci are only two, step and strep. Now have a look on the option. So my answer is straightforward streptococcus. My answer is straightforward streptococcus because only two, step and strep, are gram positive cocci. And streptococcus pneumoniae causes meningitis also, so it is fitting with my clinical scenario also. Why you all have said this thing? Why you all said this? Have you approached the same way like I have approached? Have you approached the same way like I have approached? Because otherwise all four can cause meningitis. Based on the question, if you don't see the image, you cannot tell the answer. They all cause meningitis in a child. All four can cause meningitis. But question is not in the clinical scenario. The question is in the image. But among them, the remaining three are gram negative. Only one is gram positive. The remaining three will be in red color. Only one is violet. That is the approach. Say yes if you got it. They all can cause meningitis. Meningitis बड़ी बात नहीं है. बड़ी बात है कि violet color का है. Remaining three are red color. Got my point? So that is the thing. Got it? So coming on the next question. So Sasika, that's what I'm trying to explain you, that in the filter paper they will mention कि जो filter paper है वो किसका है. So filter paper bacitracin है तो answer will be streptococcus pyogenes. और filter paper optogen है तो answer will be streptococcus pneumoniae. So they will mention कि वो जो circular disc है वो किसकी है. Image तो यही रहेगी. Got it, Sasika? है ना? So they will mention. It is not written in my diagram, but they will mention in the question. Read the next question. Culture media for streptococcus pneumoniae is sheep blood agar, in which the central depression will be there. Carom coin appearance. Okay. So we are done with this. We are done with this. Coming on the fourth. है ना, Streptococcus viridans, the second alpha. How many people are still live and alive? Only 3 points for viridians, I will tell you. Only 3 points. It's very small. Viridians के बारे में सिर्फ 3 लाइनें बोलूंगी मैं. Viridians. Let's start viridians, the second of the alpha. In the alpha, we have to study 2. है ना, pneumonia हो गया. Now I'm starting viridians. So let's start Streptococcus viridans. The first thing, it do not cause disease in healthy individual. It causes disease in opportunistic. Opportunistic means immunocompromised individual. Number 1. Number 2, it causes 2 disease. Dental caries. Very unique. Dental caries. Can you see the dant sad gaye? Dental caries. Number 1, dental caries. And number 2, heart. Endocarditis in the heart. Subacute endocarditis, not acute. So dental caries and heart make teeth and make heart. I draw the diagram of the organ. It makes me concept clear. Teeth and heart. So you will typically get a question. There is a person who is having dental caries. And I imagine this person. He is going to the dentist. He is going to the dentist for some procedure. He's having pain in the teeth. Pain in chewing or whatever. So dentist is doing the tooth extraction. The dentist is doing the root canal, the tooth extraction, some procedure on the tooth, I mean. After that, patient have some problem in the heart. Because during dental extraction, the bacteria goes inside and after that, it is involving the heart. So you get a typical question. Dentist आएगा ही आएगा उस question में. So every question have a clue. Neonate आएगा तो it will be agalacty. Dentist आएगा तो it is viridians. है ना? So take the clues. So learn that. You will say, ma'am, what is unique property here? What I am teaching you, the 2 alpha, the second alpha. How many alpha we have? We have 2 alpha. Which 2 alpha we have? We have Streptococcus pneumoniae, which I have already completed, and I am teaching you Streptococcus viridans right now. Hana? The unique properties of pneumonia is BOI, Bank of India, bile-soluble positive, optogen sensitive, inulin fermentation. The unique feature here is BOI negative. That is the only unique feature. There is no unique feature. Hana? BOI negative is की खास बात है. The unique feature, it is BOI negative. Both are alpha, na? Both are alpha. It is a Streptococcus gram-positive cocci, which is alpha. So in alpha, we have only 2 options. If BOI positive, pneumonia. If BOI negative, viridians. Tell me the disease caused by both of them. You will see, ma'am, here 2 disease, pneumonia and meningitis. Hana, here also 2 disease. Hana, here you draw the lungs, here you draw the brain. Hana, here you draw the teeth, dental caries, and you draw the heart, endocarditis. I draw the organs, it gives me better concept. Hana, so pneumonia and meningitis here, and dental caries and heart here. See the diseases. This is BOI positive, this is BOI negative. Both are alpha. Say people, yes, yes, yes. Hana, koi confusion? Hana, we are done with both alpha. We are done with both alpha. Hana, we are done with both alpha. We are done with both beta. Let's come on the last gamma. Last gamma. Can I start the last gamma? So coming on the last gamma, enterococci. ठीक है जी? Hana, the same thing is written in front of you. You can see 1 second. BOI. BOI. BOI positive, positive, positive. BOI negative, negative, negative. Hana, pneumonia, viridians. This is in pairs. This is in chains. Hana, this is lens shaped. Hana, this is like this. Hana, this is oval only. Hana, here capsule present, here capsule absent. Here carom coin present, carom coin absence. उसके अलावा the most important, BOI positive, BOI negative. ठीक है जी, है ना? So read the question, tell me the answer. After that, we will come on enterococcus. Read the question. There is a patient with rheumatic heart disease having infective endocarditis after dental extraction. Typical question. पेशेंट ने डेंटल एक्सट्रैक्शन कराया और उसके बाद एंडोकार्डाइटिस. सो डेंटल एक्सट्रैक्शन के बाद हार्ट, डेंटल एक्सट्रैक्शन के बाद हार्ट, इट हैज टू बी विरिडियंस. सो करेक्ट आंसर इज विरिडियंस ओनली. है ना? यू गेट टिपिकल क्वेश्चन. So we are done with both beta, we are done with both alpha. Lastly, I'm coming on gamma. Can I start the last gamma? Let's start with gamma. ठीक है? Gamma. Gamma में only 2 things you have to learn. The 2 unique properties of gamma. The first thing, the gamma is also in pair. Gamma is also in pair like pneumonia. So this gamma is also in pair like pneumonia. But pneumonia was flame shaped like this and capsulated. This one is circular only in pairs and a little bit angle. It is known as spectacle. Spectacle appearance. What does it known as? Spectacle appearance. ठीक है? And it is non-capsulated. यहाँ पे capsule है. Here we don't have the capsule. So it is also in pair. It is known as spectacle appearance. Spectacle मतलब चश्मा. Spectacle appearance. है ना? So typical spectacle appearance. है ना? It is non-hemolytic. पता ही हमें we are studying gamma. है ना? 2 unique properties जो कहीं और नहीं है. 2 unique property present only here, none anywhere. 2 unique properties. Number 1, it is bile esculin test positive. Mind my words, it's not bile solubility. It's bile esculin test positive. Just learn the name. Don't go in the details, just learn the name. Bile esculin test positive. Number 1. And number two, it grows in extremes. It grows in extreme. It grows in extreme. How extreme? It can grow as high as 6.5% NaCl. It grow as high as 40% bile. It grow as high as 9.6 pH. it grow as high as 60 degrees Celsius. Don't learn, just learn it grows in extreme. So learn the two things, bile is coolant and extreme. And so read the question. Bile is coolant test is positive in Enterococcus, the unique thing. Enterococcus. We are done. How many people are with me? We are done with gram-positive cocci, chapter 1, chapter 2. You have to tell me the unique things of all of them and the diseases of all of them. Can we revise together? First, smile. Everyone smile. We are done with the two biggest chapter of bacteriology. I guess 25% of the bacteriology is done. 25 or 30%. These were a very big chapter. Still, we are left with 70-75%. So all the bacteria are remaining are very small, but you got the approach. This is the approach to study. Okay. So please revise GPC. We are done with GPC. Now we will revise. After 5 minutes, we will take a break. And then after the break, we will start GNC, gram-negative cocci. So you tell me GPC का approach. You will say, ma'am, first test you apply is catalase. सबसे पहले catalase लगा लो. First you apply catalase. If it is catalase positive or catalase negative. If it is catalase positive, it's Steph. If it is catalase negative, it's trap. है ना? सबसे पहला तो ये है. सबको समझ में आया. ठीक है? After that, let's take Steph. Trap बाद में लेते हैं. Steph में the next test is coagulase. है ना? If it is coagulase positive, then what? Coagulase negative, then what? If it is coagulase positive, it's Staph aureus. If it is coagulase negative, it's cones. If it is cones, the next test you apply is novobacin. If it is novobacin susceptible, then what? Novobacin resistant, then what? Susceptible is Staph epidermidis and resistant is Staph saprophyticus. Hanji, everyone got it? Quick confusion. Staph aureus ki diseases, the 4 toxin, the 4 diseases. याद होगा ना? Cytolytic toxin causing the pus, food poisoning, toxic shock syndrome, SSSS. You remember all, I guess. है ना? यहाँ पे एक ही disease है, epidermidis, endocarditis, prosthetic wall endocarditis. यहाँ पे एक ही disease है, UTI. याद आया क्या? UTI, I have drawn the kidneys and all. UTI. Learn the unique properties and learn the diseases. This is Staph. Coming on the strap on the next page. So let's take next page. Let's take Streptococcus. Let's start with Streptococcus, the next page. ठीक है? Alpha, beta, gamma. Alpha, beta, and gamma. The 3. There is alpha, there is beta, and there is gamma. Okay? So we have 2 alpha in our syllabus. We have 2 beta in our syllabus. We have 2 gamma in our syllabus. So tell me, what are the 2 alpha? Same as Streptococcus pneumoniae and Streptococcus viridans. Here we have 2 group A, group B. Group A is Streptococcus. There are 20, but we are studying 2. Streptococcus pyogenes and Streptococcus agalacti. है ना? If I'm doing some mistake, please बताना. Gamma एक ही है. I'm sorry, by mistake I said 2. So gamma is only one. Enterococcus. ठीक है? Tell me unique properties. 3 here, 3 here, 2 here, 2 here, 2 here. Chalo. Can we start? Can we start? And then the diseases of each of them. That's it. This is the approach we study. ठीक है? Start with pneumococcus. BOE, Bank of India positive. Bile soluble, optocin sensitive, inulin fermentation. This is BOE negative. Bank of India negative. No bile solubility, no optocin sensitivity, no inulin fermentation. ठीक है जी? So coming on the Streptococcus pyogenes. I guess it is PYR positive and it's bacitracin sensitive. Am I right? इसी पे question आएगा. Can I come on agalacti? It's hippurate positive. It's CAM positive. याद आई क्या चीजें? ठीक है? Coming on the last Enterococcus. Enterococcus में it is bile esculin test positive. This was bile solubility. ये bile esculin है. Bile esculin. And it can grow in extreme. You know the various extremes. pH, NaCl, temperature, salt. ये सब extremes. Anything else you want to say? You want to ask? कुछ तो बोलो, कुछ तो सुनो, कुछ तो बोलो. Say something. You want to tell the diseases? चलो diseases और बता दो. चलो pneumonia क्या कराता है? Obviously pneumonia तो कराएगा ही कराएगा. Obviously, but it causes meningitis also. That you have to learn. Viridians, it also causes 2 diseases. The teeth, the dental caries, and the heart, subacute endocarditis. ठीक है जी? Coming on the next. Pyogenes, it causes 2 type of diseases. Suppurative, non-suppurative. Suppurative में there are 2, respiratory and skin. And non-suppurative में there are 2, rheumatic fever and PSGN. याद आया क्या? ए गैलेक्टी में तो सिर्फ एक ही होती है, सिर्फ एक ही, ओनली वन, ओनली वन, ओनली वन, नियोनेटल सेप्सिस एंड मेनिंजाइटिस. ठीक है. एंट्रोकोकस में आई फॉरगॉट टू टेल यू, यू कैन राइट, मेरे नोट्स में नहीं है, हॉस्पिटल एक्वायर्ड इन्फेक्शन. Hospital acquired infection. Can we take a break now? Okay जी, give me a thumbs up and smile. Microbiology is a very easy subject. ठीक है, so smile on this. Okay, हंसते हंसते पढ़ना है, रोते रोते नहीं. पढ़ना तो पड़ेगा ही, तो हंस के ही पढ़ लो. ठीक है, चलो. So can we take a break now? You get all the questions cleared from the 2 chapter. Any question. Any question, any question. I taught you 5 streptococcus, they are in front of you. I taught you 3 staphylococcus on the last page. So 8 bacteria still now we have done. The 3 step and the 8 strap. I'm sorry, 5 strap. So total 8 are done till now. So all 8 are written here like this. You can see all 8. Okay, let's take a break. Okay, it's 11:30. Right now we will start at 12:15. 12:15. है ना? 12:15. Break till that. Have a cup of coffee. Have something to eat. Something light. From 12:15 till 2:15 or 2:30. We will continue till 2:30. Continuously, है ना, for the next 2.5 hours or 2 hours something. ठीक है, and then we will take a lunch break. ठीक है, in the next slot we will complete gram negative cocci, gram positive bacilli. These 2 slots will be completed in the next, है ना. और after lunch gram positive bacilli. After lunch बड़ा भारी पड़ता है मुझे, इसलिए I give lunch थोड़ा सा late, है ना. चलो, anyways, so let's take a break. I'm ending it. I guess we will create a new link. ठीक है, so let's end it. Please come on time. Please come on time. 12:15 means 12:15. Bye-bye.